next students, let's start with enzymes. What are enzymes? Enzymes are proteins. All enzymes are proteins, but one exception is ribozyme, in which RNA act as enzyme. Okay, here RNA act as enzyme. Then let's do serine proteases. In serine proteases, example, chymotrypsin, trypsin, elastase, thrombin, plasmin, clotting factors 10 and 11, and PSA, prostate specific antigen. All these are examples of serine proteases. So we have to learn which amino acids are present at the active site of serine proteases. Suppose this is the structure of serine protease enzyme. Okay, so this is the active site. This is the active site of enzyme. Okay, at the active site we have 3 sites here. Okay, this is binding site. This is also binding site. Okay, so with the help of amino acids present at the binding site, the protein will bind or will hold the substrate, right? This is protein or enzyme, right? And what is this? This is catalytic site. With the help of this amino acid at the catalytic site, the enzyme will cut the protein or will cut the substrate, right? The substrate here is also a protein only. These are enzymes mainly which are cutting proteins, right? So we have to learn the amino acids present here. Here there is histidine. At the another binding site there is aspartate, and at the catalytic site, very important, it is serine. Because serine is helping in cutting, that's why name of these enzymes are serine proteases. With the help of serine, protein is cut, right? And one important point, that serine proteases have role in tumor cell migration. They have role in tumor cell migration, right? And here I can also say that active site is made up of binding site and catalytic site. Okay. Now we have to learn that these important serine proteases cut at which amino acid? That is chymotrypsin, trypsin and elastase. Questions are usually asked on these 3. So chymotrypsin will cut or cleave at carboxy terminus of large hydrophobic bulky amino acids like, can you give me an example? Like phenylalanine, tyrosine, tryptophan, right? Then trypsin will cut at the carboxy terminus of basic amino acids like lysine, arginine. Elastase will cut at the carboxy terminus of small neutral amino acids like glycine, alanine, right? Then next, let's do enzyme kinetics. In enzyme kinetics, we learn the shapes of different graphs. So for example, the first one is between velocity and enzyme concentration. In enzyme kinetics, velocity is always taken on the y-axis and the other variable is taken on the x-axis. Okay, so this graph is linear or straight line graph. This says that velocity is directly proportional to enzyme concentration. Means as the enzyme will be increasing, velocity or rate of reaction will be increasing in same proportion, right? So this was with enzyme concentration. Next is with substrate concentration. So velocity here and substrate concentration here, right? So this is called Michaelis-Menten graph. Michaelis-Menten graph. The shape of this graph is rectangular hyperbola graph, right? So this means that initially when substrate is increasing, velocity is also increasing. But in later portion of the graph, here enzyme is saturated. Enzyme is already saturated with substrate. So further increase in substrate will not increase the velocity of reaction, right? So here in initial portion of the graph, it is first order kinetics which is followed. First order kinetics, velocity is directly proportional to substrate concentration. But in later portion of the graph, it is zero order kinetics. Zero order kinetics, velocity is independent of substrate concentration, right? Now next let's see what is Km and Vmax in case of rectangular hyperbola graph. So this is Vmax, right? This is the maximum velocity which is possible for this reaction. So this is Vmax. Now if we know a point which is Vmax, we can easily take a point which is half of Vmax, Vmax by 2, right? So when you extrapolate this point on x-axis, then this value is Km. Km is Michaelis-Menten constant. So usually students make a mistake that they will say Km is equal to Vmax by 2, but no, this is wrong. What is Km? Km is that substrate concentration at which velocity is half of Vmax. Okay, I repeat, Km is that substrate concentration at which velocity is half of Vmax, right? So Km is Michaelis-Menten constant. It is not association constant, it is not dissociation constant, okay? Km is called signature of enzyme. Signature of enzyme because it is a constant value. It does not changes with change in enzyme concentration or change in substrate concentration. Okay? It is not affected by change of enzyme or substrate concentration, but it is affected by affinity between enzyme and substrate. Km is affected by affinity between enzyme and substrate, and this is an inverse relationship. Okay? So in case of competitive inhibition, affinity between enzyme and substrate decreases. So in that case, Km will increase, right? So this rectangular hyperbola graph is between velocity and substrate concentration for simple enzymes. Simple enzymes are those enzymes which have only one active site. So this is enzyme, this is active site of enzyme, right? But for regulatory or allosterically enzymes, are those enzymes which have active site. They have active site, but they also have one allosteric site where activator or inhibitor of enzyme can bind. Okay, so these enzymes are little different from the simple enzymes, and obviously they are few in number. Most of the enzymes are simple enzymes. Okay, so for allosteric enzymes, the graph between velocity and substrate concentration is having this shape. Which is this shape? This is sigmoidal or S-shaped graph, right? Then the graph for temperature and pH. For temperature or for pH. So either I take temperature on x-axis or pH and velocity on y-axis. I'm taking both of them together because the shape of graph is same for temperature and pH, that it is bell-shaped graph. Bell-shaped graph. Because this point is very low pH or very low temperature. This point is very high pH or high temperature. And enzymes get denatured in extremes of temperature or pH. That's why velocity is negligible in this area. But velocity is having good amount in this area, right? It is of significant value in this area of the graph. This is called optimum temperature and optimum pH. Optimum temperature for human enzymes, we know that it is 37 degrees Celsius, and optimum pH is between 5 to 9, right? So velocity is maximum at optimum temperature and pH, right? So this was about enzyme kinetics. Then let's do enzyme classification. Basics of enzyme classification I have covered in the starting of rapid revision only with the concepts. So now we can do the little detail. So we know that first category in enzyme classification is oxidoreductase category. Oxidoreductase. Here we have many further subcategories. One category is oxidases, and all oxidases require copper, right? Two oxidases which do not require copper, they are xanthine oxidase, sulfite oxidase. Okay, they require molybdenum and not copper. Okay, then we have dehydrogenases. Dehydrogenases, they use NAD, NADP or FAD as hydrogen acceptor. Okay, oxidases uses what thing? Oxidases uses oxygen as a hydrogen acceptor. Dehydrogenase uses NAD, NADP or FAD as hydrogen acceptor. Example, pyruvate dehydrogenase complex of link reaction, right? So we have dehydrogenases. Then we have peroxidases. Peroxidases, they use H2O2 as hydrogen acceptor. Example, glutathione peroxidase. Okay. Then next is oxygenases. Oxygenases, when this G is written here, oxygenases, these enzymes incorporate oxygen into the substrate. They incorporate oxygen into the substrate. They can incorporate one atom or two atoms of molecular oxygen. If they incorporate one atom, then they are called monooxygenases. Their another name is mixed function oxidases. Mixed function oxidases. One more name is hydroxylases. Hydroxylases. Okay. Example, enzyme phenylalanine hydroxylase, which converts phenylalanine to tyrosine. And in tyrosine, you can see one OH group present. But be careful with one point, that hydroxylases are monooxygenases, means only one O is added in this reaction. So if I ask you what is difference between molecular weight, difference of molecular weight between phenylalanine and tyrosine, the difference of molecular weight will be by 16. Okay. Do not count as the molecular weight of O also and H also. No. You will only count the molecular weight of O added, because in hydroxylation reactions only one O is added, right? Then if two atoms of molecular oxygen added, then it is dioxygenase enzyme. Example, homogentisate oxidase or homogentisate dioxygenase enzyme, the enzyme which is deficient in L-captonuria. This is a dioxygenase, right? So we are done with oxidoreductases, oxidase, dehydrogenase, peroxidase, oxygenase, and one more you can write here is reductase. Reductase will also come in this category, right? Example, glutathione reductase, which will add hydrogen and will do reduction, right? So example in transferases is kinase, which transfer organic phosphate, phosphorylase, which transfer inorganic phosphate, then aminotransferase like SGOT, SGPT, and then methyl transferase. Okay, all these are examples of transferases. Then third is EC number 3 is hydrolase enzyme. They use water to break the bond. One example is phosphatase, the enzyme which removes phosphate. And any enzyme which breaks macromolecule bond. For example, carbohydrate is a macromolecule. Any enzyme which is breaking carbohydrate like amylase, maltase, sucrase, lactase. Okay, no need to cram this. Just understand, whenever any enzyme is breaking macromolecule, then it is adding water and breaking the bond. Okay, so any enzyme which is breaking macromolecule like carbohydrates, nucleic acids, proteins, lipids. Okay, so all these will be hydrolases. Then fourth category is lyase. These enzymes can make or break a bond, but they do not require water, do not require ATP, right? Example, synthases, okay? Example, aldolase A and B. Aldolase A is in glycolysis, aldolase B is in fructose metabolism. Then simple decarboxylases, and then hydratase. Hydratase I will take on next page. Hydratase, this enzyme is very important. It belongs to EC number 4, right? And there is word written hydra. So water is added or removed. Something is happening with water added or removed. Okay, but bond is not broken. If water added and bond broken, then it is EC number 3. Okay, but in this case EC number is 4 because water is added or removed but bond is not broken. Example of such enzyme is enolase of glycolysis, fumarase and aconitase of TCA cycle, and PEP-CK of gluconeogenesis, right? Then next is EC number 5, that is isomerase enzyme. For example, mutase, intramolecular transfer, which will do intramolecular transfer. Then racemase, which will convert capital D and capital L isomer into each other. Then epimerase, which will interconvert epimers into each other, right? Then last is EC number 6, that is ligase category. Ligase, they use ATP and they make the bond. Okay, example synthetase enzyme, right? And then carboxylase enzyme, because any carboxylase uses ABC. ABC is the mnemonic, and A for ATP, B for biotin, vitamin B7, and C for carbon dioxide plus magnesium. So any carboxylase use A, B, C plus magnesium, right? ATP, biotin, vitamin B7, carbon dioxide and magnesium, right? Then next in enzymes we will do competitive inhibition, non-competitive inhibitor and uncompetitive inhibitor. In these cases, what happens to Km and Vmax? These questions are asked. So you can make this easy table and can learn here. In competitive inhibition, I told you Km is increased, right? And Vmax will be same. In non-competitive inhibition, Km is same and Vmax decreases. And in uncompetitive inhibition, both Km and Vmax decreases. The next is, sometimes electrophoretic mobility is asked. Electrophoretic mobility of isoenzymes is asked, that which has minimum and which has maximum. So I'll give you a hint how to solve these questions. For example, LDH has 5 isoenzymes, 1 to 5, okay, and we know that CK has 3 enzymes, 3 isoenzymes, 1 to 3, creatine kinase, right? So always number 1 has maximum electrophoretic mobility and the last number has least electrophoretic mobility. So in CK it will be number 1 will be having maximum electrophoretic mobility and number 3 will be having least electrophoretic mobility, right? Then next, let's do what are coenzymes and cofactors. So any enzyme is a protein, I told you, but most of the enzymes require a non-protein portion also. Non-protein portion also. Only then the enzyme is active enzyme, when it has both the protein and non-protein portion. Such active enzyme is known as holoenzyme. Protein portion is known as apoenzyme, and non-protein portion can be of two types. If the non-protein portion is organic, then it is coenzyme. If it is inorganic, then it is cofactor. Inorganic is usually a metal, right? So organic is coenzyme, right? Let's write the examples of coenzymes. One is lipoic acid, which is a coenzyme. Then is all water soluble vitamins, that is B complex and C. All water soluble vitamins are coenzymes. Then vitamin K is a fat soluble vitamin which acts as coenzyme. And then we have nucleotides which act as coenzymes. They are NAD, NADP, FAD, FMN. These are nucleotides which act as coenzymes, right? Then in the end of enzymes, what we can do is, what are the properties of enzymes? You can write, sometimes question is asked. So enzymes increase the velocity or rate of reaction, right? They increase velocity of rate or rate of reaction. They decrease the activation energy required by the substrate. They decrease activation energy, okay? But you should be very careful that what is that which enzyme is not doing. Enzyme is not changing the equilibrium of the reaction. Enzymes does not change the equilibrium of reaction, and enzymes do not change the free energy of substrate or product, right? So be careful, enzymes do not change the free energy of substrate or product. So let's move on to our next topic of rapid revision, that is amino acids and proteins. Okay, so starting with amino acids, we know that proteins are polymers of amino acids. So many amino acids join to make proteins. Okay, now amino acid, let's do the structure of amino acid. Amino means NH2 group, acid means carboxy group. Okay, so take a carbon, attach amino on left hand side, carboxy group on right hand side, H here and a side chain R here. Okay, this is general structure of an amino acid. So amino group always on left hand side, that's why, because amino group on left hand side, that's why amino acids are L amino acids. Okay, and you take this as a acid. Okay, if you take this as acid, we know that our carboxy group containing carbon is C1 and the next one is C2. Okay, so C2, that is carbon which is adjacent to functional carbon, that is alpha carbon. Okay, so in a fatty acid or in any acid, C2 is alpha, C3 is beta, C4 is gamma, like that. Okay, now amino acids are L-alpha amino acids. L means amino group is on left, alpha means alpha carbon contains amino group. So you can see here, out of these two carbons, alpha carbon has amino group, right? Then you can see that this central carbon is asymmetric or chiral carbon. So amino acids have both optical and structural isomerism, right? So all amino acids have one asymmetric carbon. Okay, but we have exceptions here. Exceptions. There is zero asymmetric carbon in case of glycine, and there are two asymmetric carbons for isoleucine and threonine, right? Isoleucine and threonine both are essential. This also you can learn here, that just take it as an idea that to synthesize an amino acid with two asymmetric carbons is difficult for body. So body cannot make it. So these amino acids are essential in diet. So they need to be coming from outside source, that is food, right? So these are essential. Now how many amino acids are essential? We have 8 essential amino acids and we have 2 semi-essential amino acids. Which are the 2 semi-essential amino acids? They are arginine and histidine. Arginine and histidine. Okay. If a question is asked that which amino acid is semi-essential and both are given arginine, histidine, then which one will you mark? You will mark arginine, because arginine is more towards essential category as compared to histidine. If this question is asked that which amino acid is essential in children but not in adults, means this amino acid needs to be supplied in diet for children, but adults can make this amino acid. Which is this one? This is histidine, right? Now in body, amino acids are present as ionized species. Carboxy group ionized to give negative charge, amino group ionized to give positive charge. So H here and R here, right? This structure has net charge 0. Net charge is 0, right? This is known as Zwitter ion, called Zwitter ion or ampholyte. Zwitter ion or ampholyte, and this structure comes out of solution, so it is precipitated out because it is insoluble, right? Solubility is because of charges. When no charge, then it is insoluble, so it is precipitated out. Okay. Now, what is Pi? Pi is isoelectric pH. Isoelectric pH is that pH at which Zwitter ion exists. Okay, I repeat, isoelectric pH is that pH at which protein or amino acid will be precipitated, because here Zwitter ion exists. Now, we all know that acidic amino acids have extra carboxy group, so they are negatively charged, and basic amino acids have extra amino group, so they are positively charged. Now, in acidic medium, if medium is acidic, in acidic medium, proteins or amino acids exist as positively charged structures. Okay, so be careful. This is a different line I have written. First, I have written that acidic amino acids have negative charge on them. But now I am saying that the medium in which amino acid is present, if that medium is acidic, then amino acid or protein will exist as a positively charged structure, right? So, in basic or alkaline medium, proteins or amino acids exist as negatively charged structure, right? Then also note down that what is the charge on cathode, anode, cation, anion, right? Cation is positive, anion is negative, cathode is negative, anode is positive, right? So be careful with this thing. Many questions are asked. Now let's start classification of amino acids. So our first category which we are going to take is aliphatic amino acid category, in which we have 5 amino acids: glycine, alanine, valine, leucine and isoleucine. Okay, so all these aliphatic amino acids are non-polar. In this, least non-polar is glycine, and most non-polar is isoleucine. Okay, now we will be taking them one by one. So first amino acid is glycine. The side chain of glycine is very simple, that is a hydrogen atom. So because it is very simple, so it can be made in body, so it is non-essential. We don't require it in diet. Okay. Then glycine is responsible for flexibility of proteins because it creates turns and bends in the structure of proteins, right? One more amino acid is proline, which creates turns and bends. So there are structure called beta turns. So in beta turns, which 2 amino acids will be present? Glycine and proline, right? So glycine is smallest and simplest amino acid. And what are the uses of glycine? Glycine is used for the formation of creatine and creatinine. It is used in the formation of glutathione. It is used in the formation of heme, and it also makes amino acid serine. So glycine to serine enzyme is serine hydroxymethyl transferase, because there is transfer of hydroxy and methyl group. Okay, so serine is OH containing amino acid which can be formed from glycine, so it is also non-essential, right? Then second amino acid in aliphatic category is alanine. Alanine can be synthesized in body, so this is also non-essential. The side chain in alanine is just a methyl group. So just a methyl group. So it is very easy to be synthesized in body. So it is non-essential. And I already told you it is most glucogenic amino acid. Mostly 3 carbon compounds are glucogenic, and this alanine amino acid is also having 3 carbons, right? Then next 3 aliphatic amino acids, valine, leucine and isoleucine. Let's take them together because they have branched chain in the side chain. So they are branched chain amino acids, and branching is a difficult thing. Just take it as an idea. Branching is a difficult thing. So it is difficult to make these 3 amino acids in body. So they are essential, right? Then we will do one disease, that is maple syrup urine disease, which is defect in the catabolism of branch chain amino acids. Okay. So when catabolism of branch chain amino acids is occurring, then the first step is transamination step, and there is formation of branch chain keto acid. Okay. Then there is second step, which is oxidative decarboxylation step in the catabolism of branch chain amino acid. This oxidative decarboxylation step requires vitamin B1. Name of enzyme will be, you know that name of enzyme for oxidative decarboxylation is dehydrogenase. So it will be branch chain keto acid dehydrogenase enzyme. Dehydrogenase complex. It is a complex enzyme. Or Branch chain keto acid decarboxylase is the another name of this enzyme, right? Now in MSUD, this step is not occurring because this enzyme is affected. So there will be increase in branch chain keto acids and branch chain amino acids. So there will be ketosis in patient and brain is affected, leading to mental retardation in these patients. And these patients have maple syrup like odor of urine. Maple syrup like odor of urine or burnt sugar like odor. That's why the disease name is maple syrup urine disease, right? Also, these patients have abnormal muscle tone because brain is affected, and if not treated, then coma and death can occur. Yes, death can occur due to MSUD, and in fact, this disease has high mortality rate, right? Then coming to aromatic amino acids. Aromatic amino acids, we have phenylalanine, tyrosine, tryptophan and histidine. Okay, so I'm taking first one, that is phenylalanine. Okay, there is phenyl ring in alanine. So very easy to make the structure. So I'm making the common structure first. Common structure made. Now methyl group added means it is alanine. Now I'm adding phenyl ring to this alanine. Okay, don't take care of the valency, just take an idea of the structure. Okay, so this is a very big kind of side chain, very big structure. Body cannot make it. So phenylalanine is essential, right? And rings, benzene ring or phenyl ring, they are always considered non-polar. So phenylalanine is non-polar, right? And in body, phenylalanine gets converted to tyrosine with the help of enzyme phenylalanine hydroxylase. It is phenylalanine hydroxylase enzyme PAH. This reaction is irreversible and it requires oxygen. Also, this reaction requires NADPH and tetrahydrobiopterin. Tetrahydrobiopterin as the coenzyme, right? So tyrosine is formed, and in tyrosine one OH group is there. So in the structure of phenylalanine, here if I add OH, that is the structure of tyrosine. Okay, now that means tyrosine is non-essential, and its polarity is in controversy because sometimes it behaves as polar and sometimes it behaves as non-polar. Due to ring, it will behave as non-polar. Due to OH group, it will behave as polar. Okay, so according to the question, you can mark whether it is polar or non-polar. Just read the question very carefully, right? Now, deficiency of this enzyme or mutated this enzyme PAH, phenylalanine hydroxylase, leads to phenylketonuria, PKU, right? Then next aromatic amino acid is tryptophan. So let's just take an idea of the structure. There are two rings in the side chain and it is known as indole ring or indole nucleus. Okay, and the word indigo means blue. Okay, so any test of tryptophan will give blue color as a positive test. Okay, so you know the idea of the structure is it is a complex structure. So obviously tryptophan will be essential, body cannot make it, and there are rings. Due to rings, it is non-polar, right? Then what are the uses of tryptophan? Uses of tryptophan is that tryptophan is used for the formation of niacin. 60 milligram tryptophan will make 1 milligram of niacin. Niacin is vitamin B3. Vitamin B3. Usually vitamins cannot be made in body, but this is an atypical vitamin which can be made in body. And another atypical vitamin is vitamin D, which can be synthesized from sunlight. Okay. Then tryptophan is also used for the formation of serotonin, and then serotonin forms melatonin. Then let's do Hartnupp's disease. Hartnupp's disease, which is autosomal recessive. In this disease, there is amino acid urea, especially tryptophan coming in urine without a corresponding increase in its serum levels. Without a corresponding increase in its serum levels, right? And here a transporter is defective, transporter which is present in intestine and kidneys for neutral amino acids, especially tryptophan. In intestine, it absorbs these amino acids. In kidneys, it reabsorbs these amino acids. So this transporter is defective. So tryptophan will be released in urine. So when tryptophan is lost in urine, then niacin is not getting synthesized in the body. There will be niacin deficiency leading to pellagra, right? Then is carcinoid syndrome. Carcinoid syndrome. It is a tumor releasing serotonin. So serotonin, we know it is synthesized from tryptophan. So too much of tryptophan is used for the formation of serotonin, that very less tryptophan available for niacin formation. So these patients also have pellagra, right? And the end product of serotonin is 5-hydroxyindole acetic acid. 5-hydroxyindole acetic acid, which will be increased in urine in these patients, right? Then next aromatic amino acid, the fourth one is histidine. Histidine is having imidazole ring in the side chain. Imidazole ring has 2 nitrogens in the ring, right? Now, histidine is polar but less polar. It is polar but less polar. It is essential amino acid, but it is semi-essential. Okay, so both properties, polarity and essentiality, lies kind of midway. Okay, but this histidine has maximum buffering capacity. Maximum buffering capacity. Okay, and due to this maximum buffering capacity, histidine is present in hemoglobin, which is a very good blood buffer. Right? Then next, let's do basic amino acids and acidic amino acids. Basic amino acids have positive charge, acidic amino acids have negative charge. So both are polar. Okay. All the basic amino acids are essential. All the acidic amino acids are non-essential. Example of basic amino acids is arginine, lysine, and histidine also we are taking in this category because it has positive charge. Okay. Now, arginine is most polar, then lysine, then histidine here. Arginine has 3 positive charges, lysine has 2 and histidine has 1. Okay. Arginine is semi-essential, histidine is semi-essential, but lysine is essential. Okay. So basically we can say all are essential in this category, right? Then acidic amino acids are aspartate, asparagine, glutamate, glutamine, right? Aspartate and asparagine are synthesized from oxaloacetate, right? Glutamate and glutamine, they are synthesized from alpha ketoglutarate. Okay, so all the acidic amino acids are non-essential because they can be synthesized from TCA intermediates, that is oxaloacetate and alpha ketoglutarate. Then next is OH containing amino acids. We have serine, threonine and tyrosine. Okay, I already told you serine is non-essential, tyrosine is non-essential, and threonine, which is having 2 asymmetric carbons, it is essential, right? Now look at these 3 questions. Whenever these 3 questions are asked, your answer is OH containing amino acid. Understand the concept? Question number 1. Which amino acid has maximum tendency to bind phosphate? OH has maximum tendency to bind phosphate, so answer will be OH containing amino acid. Okay. Then question number 2. Which amino acid is responsible for covalent modifications? Covalent modifications means phosphorylation and dephosphorylation. Okay. Again, answer will be OH containing amino acid. Then, which amino acid is involved in O-glycosidic bonds or O-glycosidic linkages? O means we need oxygen here for this bond, which is coming from O of oxygen of OH containing amino acid. Right? So, whenever these 3 questions are asked, your answer is OH containing amino acid. And if question is asked that which amino acid is for N-glycosidic bond, N stands for nitrogen here. So, here your answer is which amino acid? Asparagine. Because asparagine has CONH, amide bond, which has nitrogen in it, which will be used for nitrogen bond. Okay. Then next is sulfur containing amino acids. We have 2, that is cysteine and methionine. Okay. In cysteine, sulfur is attached to 1 carbon. In methionine, sulfur is attached to 2 carbons. Okay. Now, sulfur valency is 2. So, both the valencies are occupied in case of methionine, right? There is no free space. But in case of cysteine, there is 1 valency free. So, hydrogen is attached here. So, that cysteine has this special sulfhydryl group. Okay. Now, methionine, in methionine, sulfur is attached with 2 carbons, which is very strong bond, right? It is very difficult to make this thing attaching sulfur between 2 carbons. So, which is not done by body. So, this amino acid is essential in diet. Okay. And methionine will be converted to cysteine in body. Methionine will be converted to cysteine. So, cysteine is non-essential, right? And cysteine has sulfhydryl group. Due to sulfhydryl group, it is polar. But methionine has no sulfhydryl group. So, it is non-polar. Right? Now, for the formation of cysteine in body, what all is required? One is methionine required. Methionine will be converted to homocysteine, and then homocysteine will be converted to cysteine. Right? Then another amino acid required is serine, and vitamin required is vitamin B6. Okay? Then we will be doing amino acid, our last amino acid here in this classification. Amino acid, that is proline. This proline is not called amino acid, it is called amino acid, because here amino group is not free. Amino group is not free because this amino group is attached with the side chain, making a ring, and this ring is known as pyrrolidine ring. Pyrrolidine ring, right? So, it is a secondary amine, not a primary amine. Proline is non-polar and it is non-essential. It is non-essential. It can be synthesized from ornithine in body. Okay. So, these were 20 amino acids. Now, our 21st amino acid is selenocysteine. Selenocysteine. It is given by codon UGA, right? Then 22nd amino acid is pyrrolysine, which is given by codon UAG. Okay. We know that both these are stop codons, but by some modification, these stop codons will be giving rise to these 2 amino acids, and those modifications are co-translational modifications, not post-translational modifications. Okay. Now, precursor of selenocysteine amino acid, which is the precursor for selenocysteine 21st amino acid, it is serine. Okay. I told you glycine will be converted to serine, and then serine gets converted to selenocysteine by addition of selenium here. On the other hand, serine also gets converted to cysteine amino acid, right? Then coming to derived amino acids. So till now I told you these 22 amino acids which are coming from codons. They have a codon, right? But derived amino acids do not have a codon, but these amino acids are present in the body. Some derived amino acids are lying in the proteins. They are in the proteins, and some are not in the proteins. Okay. So derived amino acids which are in proteins are, example, hydroxyproline and hydroxylysine. They are found in collagen protein. Okay. Then the derived amino acids which are not in proteins, they are ornithine, citrulline. These are found in urea cycle. Then we have homocysteine, which is found in methionine metabolism, right? Then next we will be doing is transamination. This is the first step in the catabolism of all the amino acids, right? So what happens is that amino acid, amino group is removed, and this amino group will be taken by alpha ketoglutarate and glutamate is formed. Okay, so amino acid will be converted to its alpha keto acid, right? This is the first step in the catabolism of amino acid, and this is transamination because transfer of amino group is occurring. This step requires vitamin B6. The common amino acid in all the transamination reactions is glutamate. These reactions are reversible. Example of transaminase enzymes are SGPT and SGOT, right? In transamination, only alpha amino group takes part. Only alpha amino group can take part in transamination. But there is one exception, that delta amino of ornithine takes part in transamination, and this leads to the formation of proline, and the enzyme here is OAT, that is ornithine amino transferase, right? Then 17 amino acids, they take part in transamination, but 3 amino acids, they do not undergo transamination. We have to learn these 3 amino acids by a mnemonic, that is polythene. Polythene, write P-O-L-Y and thene. P-O-L-Y and thene. These are the 3 amino acids which do not undergo transamination. Very good mnemonic. It is very clear. You can yourself tell. PO is which amino acid? PO is proline. Okay. LY is which amino acid? LY is lysine. Okay. Thene is which amino acid? Threonine. Right. Then question is asked that what is the transport form of ammonia? Transport form of ammonia from body, from brain, and from muscles. These questions are asked. From body and from brain, it is glutamine. It is glutamine, right? And from muscles, it is alanine. Okay. So alanine is involved in Cahill cycle. Let's do Cahill cycle on next slide. So let's see this diagram. This side is liver cell, this free space is blood, and this is muscle cell. Okay. Now in fasting muscle, what happens that glucose gets converted to pyruvate, and pyruvate gets converted to alanine. Okay. The enzyme is SGPT. Okay. Now alanine gets transported via blood to liver. In liver, alanine will be again converted to pyruvate, and pyruvate will be converted to glucose by gluconeogenesis, right? This glucose will again go via blood into the muscle cell. So the cycle is complete. This is known as Cahill cycle or glucose-alanine cycle. Glucose-alanine cycle or Cahill cycle. Okay. Now let's see the full story of ammonia transport, how it occurs from peripheral cell to liver. So suppose this is peripheral cell. This is any peripheral cell of the body. This free space is blood and this is liver cell on right hand side. Okay. Now in peripheral cells, amino acids are getting catabolized. All the amino acids getting catabolized. So what is the first step? First step is transamination. Okay. Via transamination, there is formation of a common amino acid, that is glutamate. This glutamate gets converted to glutamine by free ammonia trapping here. This is free ammonia trapping here. This is also called first line ammonia trapping. The enzyme involved here is glutamine synthetase. Glutamine synthetase. Okay. Now this glutamine is the transport form in blood. So via blood it is reaching liver. Glutamine reaches liver. So there is loss of amino group from glutamine. Enzyme involved is glutaminase. And what we get is glutamate. Okay. Then from glutamate also there is removal of one amino group and alpha ketoglutarate is formed. Now these amino groups which are released in liver, they will enter into urea cycle, getting converted to urea, and urea is thrown out of the body. Now the conversion of glutamate to alpha ketoglutarate has one important enzyme to learn. This enzyme is glutamate dehydrogenase. Glutamate dehydrogenase. Dehydrogenase means oxidation. So this reaction is oxidative deamination because oxidation is also occurring and amino group removal is also occurring. Okay. This enzyme glutamate dehydrogenase can use NAD or NADP, whichever is available. Okay. This is very important enzyme and it is present in mitochondria. Okay. Now what is this term? Transdeamination. Transdeamination is coupling of transamination which occurs in peripheral cells. This is coupled with oxidative deamination which occurs in which cell? It occurs in liver cell, right? So this is transdeamination. So now moving on to next topic, that is proteins. So proteins are polymers of amino acids. So two amino acids join to make proteins. Let's see how two amino acids join here. So this is amino acid number 1 with R1 side chain. This is amino acid number 2 with R2 side chain. Let's see how these two amino acids join together. When they join, any macromolecule, when it is formed, water is removed. Okay. So this H2O will be removed to make this polymer, that is protein, and this CO and this NH, they will join. The CO and H join to make this amide bond between amino acids. So general name of this bond is amide bond. When amide bond is present in proteins, then it is known as peptide bond. Okay. Peptide bond is a strong covalent bond, and it has partial double bond character. This double bond is in trans configuration. Okay. In case of fatty acids, the double bond in unsaturated fatty acid is in cis configuration normally. Okay. And in proteins, this peptide bond is in trans configuration, right? Now next let's do structures of proteins. Proteins have primary, secondary, tertiary and quaternary structure. Okay, primary structure is sequence of amino acids all attached together in a line. Okay, now folding of primary structure will give rise to secondary structure. Secondary structure example is alpha helix, beta sheet, beta turn, right? Alpha helix is very important. Alpha helix is a helical, symmetrical helical structure, right? You need to learn many amino acids which are never found in alpha helix. These are glycine, proline, they are never found in alpha helix. Tryptophan, okay? Tryptophan because it has bulky side chain. Glycine, proline are not found because they create a bend in the structure. So alpha helix is not having any bend. It is a symmetrical helical structure, right? Then aspartate, glutamate and valine. These are also not found in alpha helix, right? Then what is tertiary structure? When you further fold secondary structure, then we get tertiary structure. And tertiary structure is fully folded single polypeptide chain. Okay, it's a single polypeptide chain. Now, quaternary structure means when more than one polypeptide chains are present in a protein, like hemoglobin has 4 chains. So, hemoglobin has quaternary structure. But myoglobin is having only one chain in it. So, we can say that there is no quaternary structure for myoglobin, or for monomeric proteins which have only one monomer, they do not have quaternary structure, right? Then let's do this table of differences between primary, secondary, tertiary and quaternary structures of proteins. The first point is bonds. So bond in primary structure is covalent bond or peptide bond or amide bond, right? Then in secondary structure, these structures are stabilized by hydrogen bonds, right? Then the bonds present in tertiary structure. In tertiary structure, there are disulfide bonds and see this mnemonic HHI, disulfide plus HHI. Like this you have to learn. What is this HHI? One H stands for hydrophobic, another H stands for hydrogen, I stands for ionic, okay? Hydrophobic, hydrogen, ionic, these bonds are present in tertiary structure. Then in quaternary structure also, same mnemonic, that is HHI. These 3 bonds are present, okay? Ionic or electrostatic is one and the same thing, right? Then next is functional activity. Primary and secondary structure, they cannot do function. They are not present in vivo also. They are breakdown products of proteins obtained in vitro, okay? Now the functional structure is tertiary or quaternary structure, right? They can do the function. Then third point, on denaturation. On denaturation, secondary, tertiary and quaternary structure is lost, okay? But primary structure is retained because of the strong covalent peptide bond that is not broken on denaturation, right? Then next point is how to detect their structures. To detect primary structure, there are 2 techniques, mass spectrometry and Edman's technique. The one I have written first, that is mass spectrometry, is best technique for primary structure, okay? Then for secondary, tertiary, quaternary structures, there are 2 techniques again, X-ray crystallography and NMR spectrometry, okay? One I have written first is best, that is X-ray crystallography is best for the detection of secondary, tertiary or quaternary structure of proteins, right? But for X-ray crystallography, as the name says, crystal, so protein needs to be crystallized. But some proteins are non-crystallizable proteins. So for these non-crystallizable proteins, best is NMR spectrometry, right? Also note down the bonds in enzyme substrate interactions. The bonds in enzyme substrate interactions are also again mnemonic HHI. Mostly these bonds are present, that is hydrogen, hydrophobic and ionic. Mostly HHI. Sometimes there is covalent bond in enzyme substrate interactions, sometimes covalent bond, and never van der Waals forces. Van der Waals forces or van der Waals interactions, right? Then let's also do the bond between protein and DNA. In protein-DNA interactions, which bonds are present? Now the mnemonic is HIV. The virus HIV, okay, that is the mnemonic. So there's only one H, and this H is hydrogen, and be careful, it is not hydrophobic. What is I? I is ionic. V is van der Waals forces, and never covalent bond here. Never covalent bond, right? So this is it with proteins topic. So next topic, students, that is lipids. So let's start with this important topic, that is lipids. Tell me the definition. What are lipids? Compounds which are not soluble in polar solvents like water. Okay, they are non-polar and they are soluble in non-polar solvents, non-polar organic solvents. Okay, like ether and alcohol, that is non-polar organic solvents. Okay, now these lipids are made up of two things, alcohol and fatty acid. Fatty acid has carboxy group, COOH group. Okay, alcohol has OH group, right? So because of OH group we know that alcohol is polar. Because of carboxy group most of the fatty acids are polar. Okay, so these two things join together to make fat or ester or lipid, and this lipid is non-polar. So this non-polar ester bond is made up of two polar things, right? And in this process water will be removed, right? To make a macromolecule, water is removed. Then let's do classification of lipids. Lipids are divided into simple lipids and complex lipids. What about simple lipids? Simple lipids have only these two components, fatty acid and alcohol. Okay, but complex lipids have one component in addition to fatty acid and alcohol, and that is called other component. Okay, so this additional other component can be phosphate or it can be a carbohydrate. Okay, if this component is phosphate, then the lipid is known as phospholipid. Okay, then it is known as phospholipid. If the other component is carbohydrate, then this lipid is known as glycolipid, right? Now this phospholipid has phosphate as the polar component, lipid as the non-polar component, so it is amphipathic. It has both polar and non-polar components. On the other hand, glycolipid, the carbohydrate portion is polar, lipid portion is non-polar, so this is also amphipathic, right? Now we will be taking all the three, simple lipids, phospholipids and glycolipids, one by one. Now let me tell you what is the alcohol. The main alcohol in lipids is glycerol. It is glycerol. The structure of glycerol is CH2OH, CHOH, CH2OH. Okay, this is a structure with 3 carbons and 3 hydroxy groups. So I will use the symbol of alphabet E and put OH on each arm. This is a kind of simplified symbol of glycerol we will be using to make the structures. Okay, so first we will be making the structure of simple lipid. So in simple lipid we take alcohol, that is glycerol. Okay, then we attach one fatty acid. Okay, one fatty acid attached. So this structure is known as monoacyl. Mono means one, acyl means fatty acid. So it has one fatty acid on glycerol. So the full name is monoacyl glycerol. Okay. In case of monoacyl glycerol, it has two OH groups, so it is polar. But this bond will be ester bond. I told you OH, that is alcohol plus fatty acid, will give rise to ester bond, and that is non-polar. So monoacyl glycerol has both polar and non-polar components. Now if we attach one more fatty acid to this structure, now we have two fatty acids. This structure is known as diacyl glycerol, having two fatty acids, right? Then if I attach third fatty acid also, then the name is triacyl glycerol, triacyl glycerol, or triglycerides, or also called neutral fat because they are non-polar. Okay. Now all the three are ester bonds here, right? Then coming to phospholipids. In case of phospholipids, we have glycerol, then we have two fatty acids attached, and at the third position there is phosphate attached. Okay, always with the phosphate some side alcohol is attached. Like this can be choline or this can be ethanolamine. Okay, if this side alcohol is choline, then the lipid is known as lecithin, which is present in lungs as surfactant. If the side alcohol is ethanol, then the phospholipid is called cephalin. Cephalin is present in CNS. Okay. So questions are asked that what are the breakdown products of lecithin? Let's say lecithin is asked. Breakdown products of lecithin. So all these bonds will be broken, and breakdown product of lecithin will be one glycerol, two fatty acids, and one phosphate and one choline. Okay. In case of cephalin, if asked, then instead of choline we have ethanolamine, right? Then what are the enzymes which break down phospholipids? Enzymes are known as phospholipases. We have A1, A2, C and D. Okay, so it's very easy. In the sequence, A1 will cut here and will release the first fatty acid. Phospholipase A2 cuts here and releases the second fatty acid. Phospholipase C cuts here before the phosphate bond. Phospholipase D cuts here after the phosphate bond, right? Then next phospholipid is cardiolipin. Cardiolipin is a complex phospholipid. It is a complex phospholipid. The structure is complex, that is glycerol is attached to 2-phosphatidic acid. I repeat, glycerol attached to 2-phosphatidic acid. And what do you mean by phosphatidic acid? Phosphatidic acid has one glycerol, two fatty acids and one phosphate. Okay. So all these things will be multiplied by 2 in case of cardiolipin, plus there is one extra third glycerol also attached here, right? So this is a complex structure. Due to its complexity, it can be sometimes antigenic in the body. Okay, it can act as antigen in the body. For example, there is a disease, antiphospholipid syndrome, right? In antiphospholipid syndrome, there are anti-cardiolipin antibodies. Which phospholipid? Cardiolipin. So there are anti-cardiolipin antibodies in the patient. There is history of recurrent abortions in the affected female, and it's a thrombotic condition. It's a thrombotic condition. So in this condition, cardiolipin phospholipid is acting as an antigen in the body, right? Now next let's do types of phospholipids. Phospholipids are of two types, glycerophospholipids and sphingophospholipid. Okay, glycero means alcohol is glycerol. Until now we have done all the glycerophospholipids. Another category is sphingophospholipid, in which alcohol is sphingosine. Alcohol is sphingosine. Okay, what is this alcohol? So to simplify, what I will do, first I will make the simplified structure of glycerol. Okay, that I have made. In this structure I will show you few changes to tell you what is sphingosine. Okay, now in glycerol there are only 3 carbons, but in sphingosine this carbon skeleton is extended. Okay, this is a long carbon skeleton, and instead of second number OH group here, it has amino group here, and it is an unsaturated alcohol. Means somewhere in this alcohol there is a double bond. So it is an unsaturated amino alcohol, right? Now let's make a sphingophospholipid. So in sphingophospholipid there is alcohol, there is fatty acid, and then there is phosphate. Okay, so the alcohol is sphingosine alcohol. Here fatty acid is attached with the amino group. When fatty acid is attached with amino group, then this bond is amide bond. Okay, amide bond, and this much structure is known as ceramide. Because it has amide bond, so it is ceramide, right? So we have taken alcohol, we have taken fatty acid. Now phosphate will be attached. So phosphate is attached here with this OH group here, and the side base is choline. Okay, the side base is always attached with the phosphate. So this is choline, and this structure is known as sphingomyelin. Okay, sphingomyelin, that is present in myelin sheath, and this is the only sphingophospholipid of concern in human body, right? So with this we are done with phospholipids. Now moving on to glycolipids. In case of glycolipids, alcohol glycerol is never present. Alcohol is always sphingosine. So we can also name them glycosphingolipids. Glycosphingolipids. Okay. So in glycolipids, phosphate is never there. So the side base or side alcohol which is attached to complete the valency of phosphate, that is also not there. Okay. So the structure is very simple. First we have sphingosine alcohol. Then we have one fatty acid. This much structure is known as ceramide. Then we have carbohydrate. Let's say glucose is there. Okay, so glucose is attached. So this is known as glucocerebroside. They are called cerebrosides, glucocerebrosides, or glucosyl ceramide. Right? Note one point that glucocerebroside, glucose form, is never found in CNS. It is only found in extraneural tissues. Right? Now, if instead of glucose we have galactose here, so sphingosine plus fatty acid plus galactose monosaccharide is used, then the structure will be galactocerebroside or galactosyl ceramide. This galacto form is always found in CNS. Right? So with this we are done with classification of lipids. Now our next topic is PUFAs. PUFA. PUFAs, that is polyunsaturated fatty acids. Okay, PUFA, PUFAs. Okay, they have 2 or more than 2 double bonds. That's why they are called polyunsaturated fatty acids. They are essential in diet because they cannot be synthesized in body. We need to take them in diet. So they are called essential fatty acids also, right? Now PUFAs have double bond. To number the double bond in fatty acid, there are two systems: omega system of numbering the double bond and delta system of numbering the double bond. Let's understand what is this. Suppose this is a hypothetical fatty acid. Okay, I have taken with 7 carbons, right? Now this is carboxy end and number starts from here. The number of the carbon of fatty acid starts from the carboxy end, right? So this is carboxy end of fatty acid and this is methyl end. This is called, the other one is called methyl end of fatty acid because there is CH3 here, methyl group. Okay. Now suppose there is a double bond between C4 and C5. Okay. If we are starting numbering from the carboxy end, then it will be given a delta number. If we are starting the numbering from methyl end, then it will be given omega number of the double bond. Okay. For example, this fatty acid, if I want to give it delta number, then it will be delta 4 because it will start, I will start from here. This is 1, 2, 3 and 4. So at 4th position double bond is there. So this is delta 4. On the other hand, if I start from omega end, then this is number 1, this is number 2, this is number 3. So this is omega 3 fatty acid. Okay. So if numbering starts from methyl end, then this is omega system of numbering the double bond, and omega system is preferred over delta system, right? So we divide PUFAs into omega 3 category and omega 6 category. So let's do 3 fatty acids in each category. So PUFAs or essential fatty acids, there are 2 categories, omega 3 and omega 6. In omega 3 category, the first one is cervonic acid. Cervonic acid is also called DHA. What is DHA? Docosahexaenoic acid. Docosa means 22 carbons. It is written here 22 carbons. Okay. Hexaenoic means, hexa means 6, ene means double bond. So it has 6 double bonds, right? And this cervonic acid is very important component of breast milk, and it is always present in breast milk, and it is required for brain development of the child, right? And health drinks are fortified with DHA these days, and this is also present in fish oil. Okay. Then alpha linolenic acid, 18 carbon and 3 double bonds. You have to learn number of carbons and number of double bonds in all these fatty acids, right? So this is present in flaxseed oil and soybean oil. The next one is timnodonic acid, 20 carbon, 5 double bonds, and this is also present in fish oil. It is also called icosapentaenoic acid. Icosapentaenoic acid. Okay. Now the precursor of this category, precursor of omega 3 category is alpha linolenic acid. Means if we are getting alpha linolenic in diet, then it can make the other 2 fatty acids of this category in our body, right? So it is precursor or prototype of this category. Then omega 6 category. First one is gamma linolenic acid, 18 carbon and 3 double bonds. This is present in oil of evening primrose, right? And then is linoleic acid, 18 carbon, 2 double bonds, present in safflower oil, right? So one thing is very easy, linolenic or linoleic, all are 18 carbons. So any fatty acid starting with L has 18 carbons. If it is smaller name, linoleic, then it has 2 double bonds. Okay. If it has bigger name, linolenic, hear the sound? Linolenic. So it has 3 double bonds, right? Easy to learn. Then the third fatty acid in this category is arachidonic acid, 20 carbon and 4 double bonds. It is present in the animal fats, right? And this arachidonic acid is important for the synthesis of prostaglandins in body, right? Now, which is the precursor of omega 6 category? This one, linoleic acid. One I have written on number 2 position. This is the precursor of omega 6 category. So it can make the other 2 of omega 6 category, right? Now, if most essential fatty acid is asked, out of all these 6, most essential is linoleic, okay? And Which is cardioprotective PUFAs if asked? Cardioprotective PUFAs, they are omega-3, right? So we are done with the topic of PUFAs. Now next coming to lipid transport. How lipids are transported in blood, because blood is polar and lipids are non-polar. Okay, so what body will do? Body is very smart. Body will put lipids in the core, and the periphery is made up of a polar component, that is protein. So this structure having lipid plus protein is known as lipoprotein. So lipids are transported in blood with the help of lipoprotein. Okay. So lipoprotein has lipid plus protein. Which lipid is present in lipoprotein? We have triglyceride, phospholipid, cholesterol, cholesterol ester, right? Triglyceride is non-polar. Phospholipid is amphipathic. Cholesterol is having one OH group, so it is amphipathic. Cholesterol ester is non-polar. Okay, so the non-polar lipids like triglyceride and cholesterol ester, they are present in the core, and the amphipathic lipids are towards the periphery. Okay, the non-polar portion of amphipathic lipid will be towards the core and polar portion will be towards outside. This way amphipathic lipid is present. For example, phospholipid will be present like this, lipid portion inside, phospho portion outside. Cholesterol will be present like this, all that is OH portion outside and the lipid portion of cholesterol towards inside, right? So this is the way these four lipids are arranged in the structure of lipoprotein. Now we will do this table of lipoprotein. There are these 6 lipoproteins we will be doing in sequence, that is chylomicron, then its remnant, chylomicron remnant, then VLDL, then its remnant, VLDL remnant, which is also called IDL. IDL full form is intermediate density lipoprotein. VLDL is very low density lipoprotein. Then LDL, low density lipoprotein. HDL, high density lipoprotein. Right? So we have to learn that which lipid is present in which lipoprotein and which protein is present. Right? Protein is also called apoprotein. Right? Now, why I am going to use this sequence? Because in this sequence, density is increasing. So chylomicron has least density and HDL has maximum density. That's why the name high density lipoprotein. Okay. Now, density is directly proportional to percentage of proteins. So maximum proteins are present in HDL, right? Density is inversely proportional to triglyceride content. So maximum triglyceride content will be present in chylomicron, right? Also, density is inversely proportional to size of lipoprotein. So chylomicron is largest in size, HDL is smallest in size, right? Now let's do this table. So from diet, the main lipid which we are having is triglyceride. Okay, this triglyceride will be coming to intestine. Now intestinal cell has to give this triglyceride to blood. Okay, so directly triglyceride cannot be going into the blood. So intestinal cell will put triglyceride in the core and the periphery will be having a protein, that is ApoB48. So this structure is known as chylomicron. So chylomicron has which lipid? It has exogenous triglyceride or dietary triglycerides, right? And which protein? It has ApoB48, right? Now chylomicron will move in the circulation and it will be giving its triglyceride to all the body parts and finally becoming smaller in size, known as chylomicron remnant. Okay. Now let me tell you one thing, very easy, that whenever you have to write remnant, you add cholesterol and lipid and you should add ApoE in the protein. So chylomicron remnant has triglyceride plus cholesterol and ApoB48 plus ApoE, right? So this was the story of exogenous fat. Now, endogenous fat, let's start the story of endogenous fat. So endogenous fat is synthesized in liver in the form of triglyceride. Again, liver has to give this endogenous fat to the other body cells. So liver will throw this fat into the blood. But liver has to do some modification because non-polar triglyceride cannot be transported in blood, right? So triglyceride in the core and ApoB100 protein in the periphery. This structure is VLDL. So it has triglyceride, but now it is endogenous triglyceride in VLDL, and VLDL is synthesized in liver. Chylomicron was getting synthesized in where? Which cell? Intestinal cell, right? Okay. So VLDL has triglyceride as the lipid and ApoB100 as the protein. Then VLDL remnant, it has TG plus cholesterol and ApoB100 plus ApoE. Okay. With the help of ApoE, all the chylomicrons are taken up by liver again, right? Now, LDL. So VLDL remnant will further lose its triglyceride and become LDL. When a point is there that all the triglycerides are lost and only cholesterol is left, then it is LDL, right? And same proteins, ApoB100 plus ApoE, right? Then HDL. HDL is also synthesized in liver. It will do reverse cholesterol transport. Now HDL has which lipid? It has cholesterol ester, right? And which protein? ApoA, C and E, right? Now, how HDL has cholesterol ester? HDL will do reverse cholesterol transport. Reverse cholesterol transport means it will take cholesterol from peripheral tissues back to liver, okay? So, when HDL goes to peripheral tissues, it will acquire cholesterol. First, it will acquire cholesterol. Then it will add one fatty acid in cholesterol. So when we add one fatty acid in cholesterol, then what we get is cholesterol ester. So this cholesterol ester will be transported to liver, right? Now we have to see that how HDL adds this fatty acid. HDL has maximum phospholipids out of all the lipoproteins, right? This phospholipid is lecithin. In the structure of lecithin, there are two fatty acids. One fatty acid will be taken from lecithin and the structure left is known as lysolecithin. Okay. This one fatty acid will be taken from lecithin and given to cholesterol. So name of enzyme is LCAT. That is lecithin cholesterol acyl transferase. Acyl means fatty acid. So fatty acid is getting transferred from lecithin to cholesterol, right? Now ApoA1 is the activator of this enzyme and ApoA2 is the inhibitor of this enzyme, right? So chylomicrons, they transport exogenous triglycerides from where to where? From intestine to peripheral tissues, right? Then VLDL transports endogenous triglycerides from liver to peripheral tissues, right? Then LDL transports cholesterol from liver to peripheral tissues, right? And HDL transports cholesterol from peripheral tissue to liver, right? So as it is doing work opposite to LDL, so this is called reverse cholesterol transport. Then I already told you about lipoprotein lipase enzyme in the starting, that this is an anabolic enzyme. So it will be activated by insulin. So what this enzyme is doing, it is converting chylomicron and VLDL into their remnants, chylomicron remnant and VLDL remnant, right? And in this process, triglyceride is getting lost from them, right? And this triglyceride is getting stored in adipose tissue, right? So that's why this enzyme is anabolic enzyme. Now, next, let's see the ligands which are present on lipoproteins so that they are taken up by liver, right? So many of the lipoproteins are taken up by liver. Suppose this is liver. So with the help of which ligands lipoproteins are taken up by liver, we have to see that. So for remnants, it is ApoE. With the help of ApoE, remnants are taken up by liver, right? For HDL, it is ApoA, right? And for LDL, there are special LDL receptors present on the liver which are doing receptor mediated endocytosis. Receptor mediated endocytosis means when LDL particle will attach to this receptor, both are taken up into the liver cell, right? Now what are the ligands here? Here the main ligand is ApoB100. ApoB100, also ApoE is the ligand here, right? Now let's do hyperlipoproteinemias. Hyperlipoproteinemias, the latest Fredrickson classification of hyperlipoproteinemias, right? So we will be making a table here: type, defect, which lipoprotein is increased, triglyceride or cholesterol, which is increased. Okay, so let's start from type 1. In type 1, the defect is in lipoprotein lipase enzyme, or ApoC2 defect, because ApoC2 is the main activator of this enzyme. So there's defect in this enzyme. This enzyme converts chylomicron and VLDL to their respective remnants. So when this enzyme will not work, then chylomicrons and VLDL will increase. Okay, so which lipid will increase? Because chylomicron and VLDL are increased, they have triglyceride. So triglyceride is increased and cholesterol is normal. Okay, so here in this disease, chylomicron increase is more than VLDL increase. So this is known as familial hyperchylomicronemia. Okay, this is the name of this type 1, familial hyperchylomicronemia, right? Then type 2A. In type 2A, there is defect in the LDL receptor or ApoB100 defect, because that is the main ligand for this receptor. Okay, so which lipoprotein will increase? Obviously LDL will increase. It has cholesterol, so cholesterol increases, but triglyceride is normal, right? And this is known as familial hypercholesterolemia. Right? Then type 2B. In type 2B, the defect is unknown. We don't know the defect, but the lipoprotein increased are VLDL and LDL both, right? Now VLDL has triglyceride, so triglyceride increases. LDL has cholesterol, so cholesterol increases. So because both triglyceride, cholesterol are increased, so it is known as familial combined hyperlipoproteinemia. Okay, familial combined hyperlipoproteinemia, right? Then the last is type 3. Type 3, that is ApoE defect. ApoE defect, where remnants will increase, right? And remnants have both cholesterol and triglyceride, so they both will increase. And this is known as dis beta lipoproteinemia, also called broad beta disease. broad beta disease, right? Then you need to know some clinical features which are having correlation with triglyceride or cholesterol, right? These will help you in question solving. So see here, if tendon xanthoma given in the question means cholesterol is increased. Okay. If eruptive xanthoma written means triglyceride is increased. If in the question milky plasma is written means chylomicron is increased. Okay. Then acute pain abdomen or acute pancreatitis mentioned in the question means triglyceride increased, because hypertriglyceridemia is the most predisposing factor to acute pancreatitis, right? Then Palmer and tuberoeruptive xanthoma means remnants are increased, right? So next topic is lipoprotein X, which is an abnormal lipoprotein. Lipoprotein X, an abnormal lipoprotein. This lipoprotein is increased in LCAT deficiency and cholestatic states, right? And this lipoprotein X is dangerous because it is rich in amphipathic lipids. It is rich in amphipathic lipids which are located towards the periphery of the lipoprotein, and it is poor in the safe neutral lipids. Okay, that's why it is dangerous. Now our next topic is fatty acid synthesis. Fatty acid synthesis pathway. Obviously it is an anabolic pathway. Any anabolic pathway occurs in cytoplasm and it is activated by insulin, and it is occurring in fed state in body, right? The starting material of fatty acid synthesis is acetyl-CoA, and there are only two enzymes in fatty acid synthesis. The first enzyme is acetyl-CoA carboxylase, which will add carbon dioxide in acetyl-CoA. Acetyl-CoA is 2 carbon. So what we get is malonyl-CoA. Malonyl-CoA is 3 carbon, right? Now this enzyme is also called malonyl-CoA synthetase. Malonyl-CoA synthetase, and this is the rate limiting enzyme of this pathway, right? So this was the first enzyme, acetyl-CoA carboxylase. Now the second enzyme will mainly use malonyl-CoA to synthesize fatty acid. Okay. It will also use one molecule of acetyl-CoA, but more molecules of malonyl-CoA will be used by this second enzyme, and the name of this enzyme is fatty acid synthase complex. Okay, fatty acid synthase complex. This is a multi-enzyme complex. There are 6 enzymatic activities in each subunit, and there are two subunits of this enzyme, right? And this enzyme, fatty acid synthase complex, will remove that carbon dioxide which was added by first enzyme, okay? And rest of the carbons will be put into fatty acids. So the extra carbon which was put in malonyl-CoA, that is removed by the second enzyme. So we say that fatty acid is synthesized from the carbons of acetyl-CoA. Okay, so we can say that both acetyl and malonyl-CoA are used in fatty acid synthesis. Okay, both will be used, but the carbons are coming from acetyl-CoA. Okay, so fatty acid is synthesized from the carbons of acetyl-CoA, because the extra carbon of malonyl-CoA is not getting added into the fatty acid. Okay. Then which molecule is the donor of carbons? Donor of carbons for fatty acid synthesis. Which molecule is mainly used for donating the carbons? Malonyl-CoA is used, right? So be careful which question is asked. If you read carefully, then you will be able to solve. Now We know that starting material of fatty acid synthesis is acetyl-CoA. Okay, acetyl-CoA. Now, fatty acid synthesis occurs in cytoplasm. So, acetyl-CoA is required in cytoplasm. But acetyl-CoA is synthesized in mitochondria via link reaction. So, how to take acetyl-CoA from mitochondria to cytoplasm via this inner mitochondrial membrane, right? So, here we need a shuttle, and that is citrate shuttle. Okay, so this is citrate shuttle on this sheet. So, be careful, it is very important. So, fatty acid synthesis occurring in cytoplasm requires acetyl-CoA in cytoplasm so that acetyl-CoA can take part in fatty acid synthesis, right? But in mitochondria, this acetyl-CoA is formed from pyruvate via link reaction. Enzyme is pyruvate dehydrogenase, right? Now, this acetyl-CoA will enter into TCA cycle. Getting combined with oxaloacetate, it is forming citrate. Now, citrate can cross inner mitochondrial membrane. That's why the name of shuttle is citrate shuttle. So, citrate reaches cytoplasm. So, with the help of lyase enzyme, it is broken down into same two constituents. One is acetyl-CoA, another is oxaloacetate. Okay. Acetyl-CoA will take part in fatty acid synthesis. Oxaloacetate, what will happen to oxaloacetate? It will be converted to malate by enzyme malate dehydrogenase. Then malate will be converted to pyruvate. Then pyruvate enters into mitochondria. Okay. So, our cycle is complete. We started from pyruvate, we have ended at pyruvate. In shuttles, always complete the cycle. Okay. Now, very important enzyme, malic enzyme, which is converting malate to pyruvate. This is malic enzyme. It produces NADPH, and NADPH is also required for fatty acid synthesis. So, I can say that citrate shuttle is providing two things for fatty acid synthesis. One is acetyl-CoA, other is NADPH, right? Then next are ketone body pathways. One is ketone body synthesis pathway, other is ketone body utilization or breakdown pathway. Okay. Ketone body synthesis occurs only in one organ, that is liver, and ketone body breakdown or utilization occurs in muscle, brain and heart, right? Ketone body synthesis is a catabolic pathway. It occurs in mitochondria. Ketone body breakdown also, it is a catabolic pathway. It occurs in mitochondria, right? But it occurs in different cells. Okay. Now, starting material of ketone body synthesis is acetyl-CoA. The first enzyme of ketone body synthesis is thiolase, which converts acetyl-CoA to acetoacetyl-CoA, right? So, this is 2 carbon, this is 4 carbon. Then, one more molecule of acetyl-CoA is added here. Enzyme is HMG-CoA synthase, which is rate limiting enzyme of ketone body synthesis, and this leads to the production of HMG-CoA. Okay. This HMG-CoA is broken by HMG-CoA lyase enzyme, and our first ketone body is formed, that is acetoacetate. So, which is the first ketone body? Acetoacetate. This is 4 carbon. Okay. HMG-CoA is 6 carbon. Then 2 carbon are removed by HMG-CoA lyase, and we are left with 4 carbon acetoacetate ketone body, right? Then acetoacetate is converted to other 2 ketone bodies. One is beta hydroxybutyrate, and here enzyme is beta hydroxybutyrate dehydrogenase enzyme. Okay. This beta hydroxybutyrate is the most common ketone body found in blood and urine of patients of ketosis or ketoacidosis, right? On the other hand, acetone which is formed, that is formed spontaneous without any enzyme. And this acetone does not act as fuel in the body. It does not provide any energy. Instead, it has a diagnostic purpose, that acetone-like odor coming from the patient means patient is in ketoacidosis, right? And also remember that in ketone body synthesis pathway, no energy is used or produced. Okay. Now, let's do this pathway of ketone body utilization or breakdown. So, acetoacetate ketone body, how it is broken down? It will be first converted to acetoacetyl-CoA. High energy CoA bond is added here. Okay. Now, to add high energy bond like CoA or phosphate, we usually apply rule of business that ATP will be used. But the rule of business is not applied for ketone body breakdown, because here ATPs are so less in the body that they're not available to get used now. Okay. So no ATP is used. Instead, this CoA is taken from succinyl CoA, and succinyl CoA will be converted to succinate. The enzyme here is thiophorase. So which is the first enzyme of ketone body utilization? Thiophorase. And very important point, that thiophorase is absent in liver. Why it is absent in liver? So that liver can never use ketone bodies. Very important point. Okay. Next, acetoacetyl-CoA is taken by enzyme thiolase, and acetyl-CoA is formed, and then acetyl-CoA will enter TCA to provide energy in cells like muscle, brain and heart. Okay. So this way ketone body is broken down to give energy, right? And this enzyme thiolase, this thiolase enzyme is common to 4 pathways. It is there in ketone body synthesis, it is there in ketone body utilization, it is used in cholesterol synthesis, it is also used in beta oxidation of fatty acids, right? So this is it with lipids. Next we are going to do molecular biology. So students, next topic is molecular biology. So let's get started. Molecular biology refers to molecules of life, and the main molecules in our cell are DNA and RNA, nucleic acids. Okay, so we will be covering that topic here. Because of this nucleic acids, one cell give rise to another cell via replication, right? So in molecular biology, we will study nucleic acids, right? Now, nucleic acids are polymers of nucleotides. Nucleic acid, example DNA, deoxyribonucleic acid, RNA, ribonucleic acid, right? They are polymers of nucleotides. And in nucleotide, there are 3 components in a nucleotide. One is nitrogenous base, other is sugar, and third is phosphate. Okay. Now, the first 2 components, N and S, nitrogenous base and sugar, this is called nucleoside, right? So nucleoside has 2 components. There is no phosphate in nucleoside. Okay. Now DNA has which nitrogenous bases? A, T, C, G. Adenine, thymine, cytosine, guanine, right? And RNA has which nitrogenous bases? A, U, C, G. So instead of thymine, it has uracil. Okay. And what is the sugar in DNA? The sugar is deoxyribose in DNA, and it is ribose in RNA. Now let's do a table. First column I will be writing nitrogenous base, then nucleoside, and then nucleotide. Okay. Now from nitrogenous base to nucleoside we add sugar. From nucleoside to nucleotide we add a phosphate. Okay. So we will be making this table. First we will be doing purines. Purines are adenine and guanine, right? Now adenine is also present in both DNA and RNA. Guanine is also present in both DNA and RNA. Okay. Now what is the nucleoside of adenine? It is called adenosine. Write sine in the end for purines, and it is guanosine. Okay. And nucleotide, in nucleotide we add phosphate. So AMP is adenosine monophosphate, ADP, ATP. Okay, diphosphate and triphosphate. So AMP, ADP and ATP have ribose sugar in them. Now we also have deoxy AMP, deoxy ADP, deoxy ATP in the cells, right? Similarly, we have GMP, GDP, GTP and deoxy forms. Deoxy GMP, deoxy GDP, deoxy GTP. Okay. Now always for the full form of nucleotide, use the name of nucleoside. So GMP will be guanosine monophosphate and not guanine monophosphate. Okay. So this was purines. Then is pyrimidine. Pyrimidine, cytosine, uracil and thymine. Okay. We know that uracil is only in RNA, thymine is only in DNA. Okay. Now, for the nucleoside of pyrimidines, write dene in the end. It is cytidine, uridine, thymidine. Okay. So, nucleotide of cytosine is CMP, CDP, CTP and deoxyforms. Deoxy-CMP, deoxy-CDP, deoxy-CTP, right? Then uracil is only in RNA, so we only have UMP, UDP and UTP. There are no deoxyforms, okay, because it is only in RNA. Then for thymine, we have deoxy-TMP, deoxy-TDP, deoxy-TTP, only the deoxyforms because thymine is not there in RNA, okay. So tell me the full form of deoxy-TMP. It will be deoxythymidine monophosphate, right? Now next let's do formation of deoxyribose sugar, because first in body ribose sugar is formed via HMP pathway. We know ribose 5-phosphate is formed, right? Now riboform gets converted to deoxyriboform, which is required in DNA, right? Note here that this conversion only occurs at diphosphate level. That is, when it is ribonucleoside diphosphate, then at this level it will be converted to deoxyribonucleoside diphosphate, right? So ribose sugar, so here ribose sugar is getting converted to deoxyribo, so there will be removal of one oxygen and this is reduction. So enzyme is reductase. It is ribonucleotide reductase, because this full thing is a ribonucleotide having nitrogenous base and sugar and phosphate, okay. So the name of enzyme is ribonucleotide reductase. So this is getting reduced. On the other hand, thioredoxin is getting oxidized. Thioredoxin is getting oxidized, right? Now again we have to replenish the reduced thioredoxin form. So here we have NADPH. NADPH as the ultimate donor of hydrogens, and name of enzyme is thioredoxin reductase, right? This enzyme thioredoxin reductase is a selenoprotein, means it has selenocysteine amino acid at its active site, that is the 21st amino acid, okay. So ribo to deoxyribo conversion occurs at the level of diphosphate, okay. Then uracil, that a pyrimidine, gets converted to other two pyrimidines, that is thymine and cytosine. So from U to T conversion, here this occurs at the level of monophosphate, and U to C conversion occurs at the level of triphosphate, okay. So first let's look at this conversion, U to T conversion. So U to T conversion occurs at the level of monophosphate, means when it is UMP, then it will be converted to TMP, and here methyl group is added, and this methyl group is obtained from tetrahydrofolate, and enzyme is thymidylate synthase, okay. Synthase means no energy is used here, okay. Then next is U to C conversion. This occurs at the level of triphosphate, means UTP gets converted to CTP. Here from U to C, one amino group is added. This amino group is obtained from glutamine, and the name of enzyme is CTP synthetase. Synthetase means ATP is used, right? Then let's do an idea of the structure of purines and pyrimidines. So purines are adenine and guanine. The structure of purine is one hexacyclic ring joined to one pentacyclic ring, right? So one hexacyclic ring joined to one pentacyclic ring. So here if amino group is present, just look at this position at the top of hexacyclic ring. If amino present, it is adenine. If amino not present, it is guanine, right? Then we have pyrimidines, cytosine, uracil and thymine, okay? In case of cytosine, single ring and amino group present here. In case of uracil, this single hexacyclic ring, no group present. In case of thymine, single hexacyclic ring with methyl group present here but no amino group. These are the structures of purines and pyrimidines, right? Now you have to learn the sources of nitrogens and carbons of purines and pyrimidines. So look at this picture. This is the hexacyclic ring and this is pentacyclic ring joined to it. This is the structure of purine, right? In case of purine, this is N1 coming from aspartate, okay? N1 coming from aspartate. Then N3 is and N9. There are N3 and N9 coming from glutamine, okay? Then N7 coming from glycine. Also C4 and C5 coming from glycine, right? So nitrogens are done. Then C2 carbon is from tetrahydrofolate, okay? Then C8 is also from tetrahydrofolate, but C6 is from carbon dioxide, okay? So which amino acids are the sources of nitrogen and carbon in case of purine? We have aspartate, we have glutamine and glycine, okay? Aspartate, glutamine, glycine, these three are involved. But in case of pyrimidines, this is single ring, hexacyclic ring, right? So here N1 is from aspartate. This is similar. In purine also N1 is from aspartate and there is N3 from glutamine. In purine also N3 is from glutamine. So N1 and N3, they are same, right? Now carbons, C4, 5, 6. C4, 5, 6 are from aspartate. So aspartate has a very good role in pyrimidines. N1, C4, C5, C6, all are from aspartate, right? And then we are left with C2. C2 is from carbon dioxide in pyrimidines, right? And in thymine pyrimidine, there is one extra methyl group on this side, that is from tetrahydrofolate. I already told you, right? Now let's write products of catabolism of purines and pyrimidines. Products of catabolism, that is when they are broken down, then what we get. When purines are broken down, they are just converted to another purine like ring, that is uric acid, which is soluble in water and thrown out of body, okay? But in case of pyrimidines, pyrimidine ring is broken down into pieces, so there are more than one products of hydrolysis or catabolism. In case of all the pyrimidines, it is ammonia, carbon dioxide and beta-alanine, okay? But one thing is different for thymine. In thymine, ammonia and carbon dioxide are same, and another product is beta-amino isobutyrate and not beta-alanine, okay? Then next is synthesis of purines. There are two pathways. One is de novo pathway, starting from taking each and every raw material one by one. So it is a very lengthy pathway. 15 to 20 steps will occur. High energy consuming pathway. And the rate limiting enzyme of this pathway is PRPP, glutamyl amidotransferase. Okay, so this is de novo pathway. On the other hand, we have one small salvage pathway. Salvage pathway uses less energy because it has only one step. Okay, the purines which are getting catabolized, they are taken up back into the body again by salvage pathway. Okay, And the main enzyme of this pathway is HGPRT. HGPRT is, full form is hypoxanthine guanine phosphoribosyltransferase, right? So let's look at this slide of purine catabolic pathway. Here we will also look at salvage pathway. Okay, because when purines are getting catabolized, there only salvage pathway exists to save some of them back into the body again. Okay, so purine catabolism, AMP and GMP going into catabolic pathway. First, phosphate is removed from them, so we get nucleoside, adenosine and guanosine, right? Then sugar is removed. So from guanosine, we get guanine nitrogenous base. Now on adenosine side, there is one extra step, adenosine to inosine. Inosine is another nucleoside. Here there is removal of amino group from adenosine to inosine, and enzyme is adenosine deaminase ADA, right? ADA. This ADA enzyme is increased in the patient of tuberculosis, right? And if this ADA enzyme is decreased, then those patients will be having SCID, that is severe combined immunodeficiency, because ADA enzyme is very important for both B and T lymphocytes. Okay, for both B and T lymphocytes it is important. That's why deficiency will lead to SCID. Now let's move further. We have reached to inosine. From inosine, sugar will be removed. Inosine is a nucleoside, sugar removed, and we get the nitrogenous base hypoxanthine, right? Now both hypoxanthine guanine taken by enzyme xanthine oxidase, converting them to xanthine. Then again xanthine oxidase enzyme will convert them to uric acid, and this xanthine oxidase is the rate limiting enzyme of purine catabolic pathway, right? Now uric acid will be converted to allantoin by enzyme uricase in non-primate mammals, right? So this was catabolic pathway. Now what is salvage pathway? At the level of nitrogenous bases, as they're going into catabolism here, so let's say 10 nitrogenous bases are going into catabolism, but what will happen? Only 5 will be allowed to form uric acid. Remaining 5 will be again taken up back into the body and converting them to their nucleotides, and this is salvage pathway. They are again getting saved in the body, right? So hypoxanthine will make IMP, inosine monophosphate. Guanine will make GMP, that is guanosine monophosphate. Okay, and here PRPP is getting added and pyrophosphate is removed. Okay, and this common enzyme is HGPRT, right? Now complete deficiency of HGPRT is Lesch-Nyhan syndrome. In Lesch-Nyhan syndrome, patient has gout, because when this pathway is not occurring, all the purine nitrogenous bases are getting converted to uric acid. Okay, this will lead to hyperuricemia. And second is self-mutilation behavior of the patient. Self-mutilation, that is patient will do self-biting, self-harming. This is a neuropsychiatric behavior. This is because of increased PRPP, which is a neurotoxic compound, right? So this was complete deficiency. Now partial deficiency of HGPRT leads to Kelly-Siegmiller syndrome. In this, patient has only gout, no neurological problem, right? So we are done with purines. Now pyrimidine. Pyrimidine synthesis. Pyrimidine synthesis pathway's first step is similar to urea cycle step, but it is occurring in cytoplasm, right? So 2 ATP plus carbon dioxide plus source of nitrogen is glutamine here, right? Leading to the formation of carbamoyl phosphate. Enzyme is CPS2, that is carbamoyl phosphate synthetase 2. Okay, CPS1 is in urea cycle. Okay, now carbamoyl phosphate will be converted to orotic acid or orotate. Okay, now orotic acid will be converted to OMP, which is a nucleotide. Okay, so what will be the full form of OMP? Orotidine. Dene, because for pyrimidines we write dene in the end, orotidine monophosphate. Okay. Now from nitrogenous base orotate to OMP, we have to add PRPP, phosphoribosyl pyrophosphate, right? So enzyme is OPRT, that is orotate phosphoribosyl transferase. Okay. Now next enzyme is decarboxylase and UMP is formed, uridine monophosphate. Okay. Now I already told you UMP will be converted to TMP at the level of monophosphate, U to T conversion, at the level of triphosphate, U to C conversion. Now see here, OPRT and decarboxylase, they are bifunctional enzymes, means both these enzymatic activities are present on a single enzyme or single protein. When this enzyme is affected, then the patient will be having orotic acid increased, that will be coming in urine, orotic aciduria, right? In orotic aciduria, patients have megaloblastic anemia and growth retardation. Treatment is giving uridine, giving only one pyrimidine, that is uridine, because U will be converted to C and T and other pyrimidines can be synthesized in body, right? Now next, let's see the structure of ribose and deoxyribose sugar. So ribose sugar is furanose form, oxygen at the top of the ring. So this will be C1, C2, C3, C4 and C5 is out of the ring. Now at C1 there is HOH, at C2 there is HOH, at C3 also HOH, at C4 H here and at C5 CH2OH, right? This is the structure of ribose. Now at 2 number position, if oxygen is removed, then this is called 2-deoxyribose, which is present in DNA, right? Now if oxygen is removed from number 2 position and number 3 position both, then it is called 2-3-dideoxyribose. If this 2-3-dideoxyribose is used, which is an abnormal nucleotide, then DNA synthesis will stop, right? Then let's do some basics of chromosome. In human somatic cells, they have 2n, that is diploid configuration, that is 23 pairs of chromosomes are there, in which one pair is of sex chromosome and other 22 are autosomes. Okay, but in case of germ cells are haploid, that is n, so they have 23 chromosomes. Okay, now let's do types of chromosomes based on the centromere location, right? The centromere is the middle constriction, the central constriction in the chromosome, right? And the ends of chromosomes are telomeres. Okay, so if the centromere is exactly in the middle, M for middle, M for metacentric, these are called metacentric chromosomes. P arm and Q arm will be of equal size, right? And these are considered primitive chromosomes. Only chromosome number 1 and 3 are metacentric in humans. Okay, then if the centromere is between middle and end, then it is submetacentric chromosome, which is the most common chromosome found in humans. P arm is short and Q arm is long. So majority of human autosomes and X chromosome are submetacentric. Okay, then if the centromere is very close to the end, it is acrocentric. Very short P arm will be there. So Y chromosome is like this, and some autosomes, only 5 autosomes are like this, that is 13, 14, 15 and 21, 22. Okay, then the last one is telocentric, which does not exist in humans, that is centromere is at the end, means centromere and telomere both are combined. Now what are bar bodies? Next topic, bar bodies. Bar body is the inactive condensed X chromosomes. X chromosome. Okay. Now, how many bar bodies are present? Number of bar body is equal to number of X chromosome minus 1. Okay. 1 less than the X chromosome. So in a normal female, 2 X chromosomes are present. So 1 bar body will be there. Right. So see this table. Normal male, XY genotype. So no bar body. Normal female, XX genotype. So 1 bar body. Turner syndrome is a female with no bar body because genotype is XO. So there is no bar body, right? Then Klinefelter syndrome, the phenotype is of a male. So this is a male with bar body because the genotype is XXY. So there are 2 X chromosomes. So 1 bar body will be there. Usually males do not have bar body, but this is a male with a bar body, right? Then super female, 3 X chromosomes are there in the genotype. So there will be 2 bar body and the phenotype is of a female. So this is a female with 2 bar bodies, right? Then absorption of UV light by nucleic acids. So the conjugated double bonds in the rings are there. Due to these conjugated double bonds, there is absorption of UV light. So in DNA, it is because of nitrogenous bases. And DNA absorbs UV light at 260 nanometers, right? You have to learn this. Then proteins and amino acids absorb at 280 nanometers, and this is because of aromatic amino acids. And maximum absorption will be by tryptophan because tryptophan is the only aromatic amino acid having 2 rings. Okay. Then NAD, NADP absorb UV light at 340 nanometers. And porphyrins absorb at 400 nanometers. The absorption band of porphyrin is known as Soret band, right? Now, let's do the bonds in DNA, right? So, DNA is double-stranded and we know that RNA is single-stranded, okay? So, the backbone is made up of sugar and phosphate, right? Sugar is attached to nitrogenous base, and in between the nitrogenous base, there are hydrogen bonds. They can be double or triple. So this is hydrogen bond. Between A and T, there are 2 hydrogen bonds. Between C and G, there are 3 hydrogen bonds. Now, this bond between N and S is beta N glycosidic bond. Beta N glycosidic bond, which is between, this bond is between C1 of sugar and N9 of purine and N1 of pyrimidine, and it is a covalent bond, right? Then the bond between sugar and phosphate is 3-5- phosphodiester bond. This is also a covalent bond, right? So these are 3 bonds in RNA: hydrogen bonds, phosphodiester bond, glycosidic bond. And in RNA, because RNA, so these are 3 bonds in DNA: hydrogen bond, phosphodiester bond and glycosidic bond. But in RNA, because RNA is single-stranded, so there are no hydrogen bonds, others will be there. So DNA is double-stranded in both prokaryotes and eukaryotes. In prokaryotes, it is circular double-stranded. There are no introns in prokaryotes, right? And in eukaryotes, it is double-stranded linear. It is linear double-stranded, and eukaryotes have introns. Due to the presence of introns, mutation is prevented in eukaryotic DNA, right? But in eukaryotes, mitochondria is there. Mitochondria is same like prokaryotic DNA. Although mitochondria is present in eukaryotes, but the DNA of mitochondria is same like prokaryotes, but it is circular double-stranded with no introns. So there are more chances of mutation in mitochondrial DNA as compared to the eukaryotic nuclear DNA, right? Then what is palindrome? Many questions are asked on palindrome. Palindrome is same sequence same sequence on both strands. For example, I'll give you two options. Tell me which is palindrome. Which is palindrome? Many students will mark B here, but no, B is not palindrome, A is palindrome here. How? See, palindrome is present in DNA and DNA is double-stranded. So, we have to check both the strands. See, the definition also says both strands. So, TAAT written means, obviously this is 5 dash on left side and on right side it is 3 dash. Okay. When direction is not mentioned, then obviously left is 5 dash. Okay. So, other strand will be 3 dash here and 5 dash here. So, write the complementary sequence A, T, T, A. Okay. Now, read the upper strand. You always read in 5 to 3 direction. So, read the upper strand, it is TAAT. Read the lower strand, it is ATTA. So, they are not same. It is not a palindrome. But GGCC is a palindrome. GGCC. So, obviously 5 dash here, 3 dash here. So, for other strand, 3 dash here and 5 dash here. So, in front of G, there will be C. For C, there will be G. So, I have written the complementary sequence. Now, read the upper strand, it is GGCC. Read the lower strand, it is GGCC. Read always in 5 to 3 dash direction. Okay. Now, next coming to DNA replication. For DNA replication, we will be doing enzymes in sequence. So, first enzyme will be separating the strands, that is helicase. Helicase causes strand separation. It will cause strand separation. This enzyme uses ATP for strand separation, and when it is separating the strands, then it creates supercoiling ahead in the DNA. So, it creates positive supercoils, right? Then, second enzyme, topoisomerase. Topoisomerase relieve the positive supercoil because only then the replication fork can move forward. Okay. So, first enzyme creates supercoils, second enzyme release or relieve positive supercoils, right? Then, third enzyme is single strand DNA binding proteins. Single strand DNA binding proteins. They prevent re-annealing of the two strands, right? So, these three together are called unwinding proteins. They do the work of unwinding the two DNA strands, right? So, unwinding work is done. Now, what next? Fourth will be primase. Primase enzyme will synthesize RNA primers. It will synthesize RNA primers, right? We require one primer on leading strand. One primer is required for leading strand and multiple primers are required on the lagging strand. Okay. Now, what is leading and lagging strand? Suppose this is replication fork. Now, one primer and full strand is synthesized in one stretch. This will be leading strand. Okay. On the other side, one primer, a small fragment, primer, small fragment. This is lagging strand, and the small fragments are known as Okazaki fragments, right? So, one primer for leading strand, multiple primers for lagging strand, right? So, next enzyme in our sequence is fifth, that is DNA polymerase 3. DNA polymerase 3 is the main DNA polymerase enzyme. It is called DNA dependent DNA polymerase, right? So, this enzyme will make leading and lagging strand, right? Then our sixth enzyme is DNA polymerase 1. DNA polymerase 1 will remove these RNA primers. This enzyme will remove RNA primers from both the strands, leading and lagging strand. And then it will fill the gap with DNA. After removing primers, gap will be left. The same enzyme DNA polymerase 1 will fill the gap with DNA, but only on the lagging strand. On the lagging strand. Okay. The gap on leading strand will be still left. Okay. So what will be seventh enzyme? Seventh enzyme is DNA ligase, which will join the fragments. It will join the fragments. For that, it will use ATP. It will create 3-5- phosphodiester bond, and it works on the lagging strand because there is nothing to be joined on leading strand, right? So let's do this table of prokaryotic and eukaryotic DNA polymerase enzymes, right? First is DNA polymerase 1. So this number terminology 1, 2, 3 number terminology is used for prokaryotic DNA polymerases, and this alpha, beta, gamma, delta terminology is used for eukaryotic DNA polymerases, right? So let's start. The first one is DNA polymerase 1. It remove primer and fill the gap. I told you. Okay. Now in case of eukaryotes, the enzyme is different. For nucleus, these enzymes are RNase H, FEN1, that is flap endonuclease, and minor role of delta polymerase. For mitochondria, only RNase H and FEN1 and no role of delta polymerase. Then DNA polymerase 2, this is involved in proofreading and repair. Okay. On next slide, we will understand what is proofreading and repair. Now this was in prokaryotes. In eukaryotes, beta polymerase for repair, gamma polymerase for mitochondrial DNA synthesis, right? Then DNA polymerase 3 will make leading and lagging strands both. But in eukaryotes, epsilon for leading strand, delta for lagging strand. Okay. Then primase, that is primer synthesis, that is done by DNA G protein in prokaryotes and alpha polymerase in eukaryotes. Okay. This table is very important. You have to learn it. Few mnemonics I will give you here. So mitochondrial DNA synthesis, gamma. Okay, gamma. M for mitochondria, and there are two M in gamma polymerase. Gamma polymerase, right? Then when you write leading, make a big E here in leading. So epsilon polymerase for leading strand. Then write lagging. Instead of G, make like this. In lagging polymerase, when you write lagging, so you can see delta here. So lagging strand is made by delta polymerase. Easy to learn this way. Then let's do proofreading and repair. Now what is proofreading? Proofreading is correction during synthesis. Means proofreading occurs during S phase of cell cycle, right? S phase of cell cycle. Then repair means correction after synthesis. Once synthesis is done, after that if some correction is occurring, that is repair. Okay. And repair mostly occur in G1 phase of cell cycle. Okay. Then enzymatic activity. Proofreading enzymatic activity is 3 to 5 exonuclease activity. For repair, mostly endonuclease, but 5 to 3 exonuclease activity is also possible. Then enzyme for prokaryote, for proofreading DNA polymerase 2, for repair DNA polymerase 2, right? Enzyme in eukaryotes. All DNA polymerases can do proofreading except alpha and beta polymerase, and repair enzyme in eukaryotes is mainly beta polymerase and minor role of epsilon polymerase, right? So I told you 7 enzymes in sequence for DNA replication, right? So because there are only 7 enzymes and a gap is left with each replication, so with each replication some DNA will be getting lost. So therefore somatic cells have limited number of divisions. After n number of divisions, somatic cells will die because with each replication some DNA getting lost, and a time will come when there is very less DNA, that cell cannot survive. Okay. This is known as telomere shortening. Telomere shortening, because if this is a chromosome, ends of chromosome known as telomeres, right? Here there is a sequence TTA GGG repeated n number of times. Okay, so with each replication, some portion is getting lost from the telomere, known as telomere shortening, and this is responsible for aging and death, which is a normal natural phenomena. So this was about somatic cells, but in germ cells and stem cells, they have infinite divisions. Right? How? Because in these cells there is one extra enzyme, 8th enzyme, that is telomerase enzyme. This enzyme telomerase has a protein portion which acts as enzyme, and it has RNA portion which acts as template. Taking this RNA as template, DNA is synthesized on it. Okay, DNA is synthesized. The gap which we need to fill, that gap's DNA is synthesized like this. So this enzyme uses RNA as template, so it is RNA dependent, and this enzyme makes DNA on it, so it is DNA polymerase. So it is RNA dependent DNA polymerase, or also called reverse transcriptase, right? And this is not a ribozyme, because ribozyme means RNA having enzymatic activity, but here RNA is not having enzymatic activity. Instead, RNA acts as template, right? And this telomerase activity increases in cancer. It increases in cancer and it decreases with aging, right? Then next, let's do this table of repair. Okay, we have 3 types of repair: nucleotide excision repair, which occurs in G1 phase of cell cycle; base excision repair, that also occurs mainly in G1 phase; mismatch repair is one repair which occurs in G2 phase of cell cycle. Okay, now first is nucleotide excision repair. The cause is UV radiation. Whenever UV radiation damage occurs, then the damage which occurs in humans is thymine dimers get formed, TT dimers, but it is corrected by nucleotide excision repair. And if in some person it is not corrected, then which disease will occur? Xeroderma pigmentosa. Okay. Next is base excision repair. Damage is C to U conversion. In DNA, right? So U, that is uracil, is not found in DNA, so it can be corrected by base excision repair. What is the cause? Cause is spontaneous or heat or infrared rays, viral infection, or nitrous oxide chemicals. And the disease which can occur is MUT-YH associated polyposis, right? And the last one is mismatch repair. The damage is a mismatched base, cause is proofreading error, and the disease which can occur, if this repair does not occur, then the disease which can occur in human is HNPCC, hereditary non-polyposis colorectal cancer, right? So students, our next topic is transcription, right? So transcription is synthesis of RNA on DNA. So taking DNA as a template, RNA is synthesized on it. So this enzyme is DNA dependent RNA polymerase. Okay, DNA dependent RNA polymerase is the enzyme of transcription. But in replication, primase enzyme, which is synthesizing RNA primer, that enzyme is also DNA dependent RNA polymerase. Okay, so there is one RNA polymerase in replication also, right? Now this enzyme RNA polymerase has various subunits. Okay, it has alpha and omega, which are required for the assembly of this enzyme, right? Then it has beta, which is the main catalytic subunit. Okay, then there is one beta dash, which is for template binding. What is the template for transcription? Template is DNA. So with the help of beta dash subunit, this will bind with template. Okay. Then the last one, sigma subunit, sigma, which is for initiation, because it will recognize the promoter on the DNA and it will initiate transcription. Right? Then there are various types of RNA polymerases in eukaryotes. In prokaryotes, there is a single RNA polymerase only, but in eukaryotes, there are types. So let's do these types. Type 1 RNA polymerase will synthesize all the rRNAs except 5S rRNA. Right? Then type 2 will synthesize mRNA, miRNA, that is microRNA, then inc RNA. INC inc, what is this? Stands for long non-coding. Okay, the short form is INC, but there is nothing from I. The full form is long non-coding RNA. Okay. Then few SN and SNO RNAs are from type 2. Next, coming to type 3 RNA polymerase, it will synthesize 5S rRNA, tRNA, and few SN and SNO RNAs are from type 3. Okay. Then in eukaryotes, we have mitochondria. So there is a different mitochondrial RNA polymerase for the synthesis of mitochondrial RNA. Right? Then we will understand which is template strand and non-template strand in transcription. So for transcription, the two DNA strands are unwinded. Okay. Let's say this is number 1 strand, this is number 2 strand of DNA. Right? Now suppose there is gene on number 1 strand. So the strand which is having the gene, that will be the template strand on which RNA will be getting synthesized. Okay. This template strand is also known as antisense strand or non-coding strand. Right? And this is also called minus strand. Minus strand. Okay. So remember this thing, always the strand of DNA where gene is present, that is template or antisense or non-coding or the minus strand. Okay. But the other strand of DNA, this number 2 here, this is non-template strand. It is non-template strand. It is coding strand because the codons of this strand of DNA matches with the RNA. Right? Just that there will be U in RNA and T in DNA. That difference will be there. Then it is also called a sense strand because the new RNA and this non-template strand of DNA have same sense of direction. So this RNA will be 5' to 3'. Non-template strand is also 5' here and 3' here. Template strand will be 3' here and 5' here. Right? So non-template strand is called sense strand. It is also called plus strand. Right? Then next we will do ribozyme. Ribozyme is RNA acting as enzyme. So we have various ribozymes. First is 23S rRNA in prokaryotes and 28S rRNA in eukaryotes. This acts as ribozyme. The name of ribozyme activity is peptidyl transferase. Peptidyl transferase enzymatic activity. This enzyme is involved in elongation and termination in translation. Right? Then second is ribozyme having role in splicing, that is snRNA, small nuclear RNA. Okay? snRNAs type U1, U2, U4, U5, U6. Okay? So it is from 1 to 6, but 3 is not there. Okay? So why alphabet U is used? Because these snRNAs are rich in uracil. They are uracil rich and they help in splicing. Splicing is removal of introns. Removal of introns. Okay? And in splicing, snRNA combine with proteins and make SNRPs. snRNPs. SNRPs are small nuclear ribonucleoprotein particles. Ribonucleoprotein particles, right? Then third example of ribozyme is ribonuclease P. Ribonuclease P, and this ribozyme helps in cleaving the tRNA. It cleaves tRNA. Okay, so these all were ribozymes, but be careful, these two are not ribozymes. Telomerase is not a ribozyme and RNase H is not a ribozyme, right? Then we will do the next topic, that is codons. Codons are nucleotide triplets. Nucleotide triplets, that is 3 bases make 1 codon. 3 bases make 1 codon, and total 4 bases are used to make codons, that is A, U, C and G. Okay, so what is the combination possible is 4 to the power 3, that is 64 codons are possible. Okay, once a question was asked that if 4 bases make 1 codon, instead of 3 bases making 1 codon, if 4 bases make 1 codon, then what is the combination possible? Then your answer will be 4 to the power 4, that is 256 codons will be possible in this case. Okay, but this is not the case. That was just a hypothetical question asked. So we have 64 codons, out of which 3 are stop codons, that is UAA, UAG and UGA. Stop codons means they will not give any amino acid, right? They will not code for any amino acid. So we have 61 codons for approximately 20 amino acids. Or we can say that for each amino acid we have approximately average 3 codons, right? So there are more than 1 codon for each amino acid, and this property is called degeneracy of codons or redundancy of codons. Degeneracy, right? So this is degeneracy, but 2 amino acids do not show degeneracy. 2 amino acids do not show degeneracy. They are methionine. Methionine has only 1 codon, that is AUG. This is also the first amino acid which is placed in the translation, and this AUG is the initiation codon, right? So methionine has only 1 codon for it, and the other amino acid is tryptophan, that is also only having 1 codon. Then next topic is translation. Translation. In translation, activation of amino acid, that is charging the amino acid with the tRNA, that occurs first, and then initiation of translation, then elongation, and then termination of translation occurs. Okay, this point is very important, activation of amino acid. Here amino acid gets attached to its tRNA, and the enzyme is amino acyl tRNA synthetase. Amino acyl tRNA synthetase. Why synthetase? Because it is using ATP. ATP gets converted to AMP here. So this is equivalent to 2 ATP used here, right? And amino acyl tRNA is synthesized. This enzyme is the only point of proofreading in translation. It is the only point of proofreading in translation, means if some wrong amino acid gets attached to tRNA, then this enzyme will immediately correct that thing. Okay, but if something wrong occurs in initiation, elongation or termination, then that cannot be corrected, because this is the only point of proofreading of translation, right? The correction can occur only here in this step. So they also say that this enzyme is responsible for fidelity or accuracy of translation, right? And this enzyme has 20 isoenzymes, one for each amino acid, right? Now let's look at the factors required in translation for prokaryotes and eukaryotes. So for initiation, there is initiation factor 1, 2, 3 in prokaryotes, and in eukaryotes there is eukaryotic. Small e stands for eukaryotic here. Eukaryotic initiation factor 1, 2, 3 and 4F. This 4F is the rate limiting factor here. It is a very important rate limiting factor in the initiation. Then in elongation, EF, TU, that is elongation factor, TU, TS and G in prokaryotes, and in eukaryotes it is eukaryotic elongation factor 1 alpha, 1 beta, 1 gamma and elongation factor 2, right? Then for termination there are 3 releasing factors in prokaryotes, but there is single eukaryotic releasing factor in eukaryotes, right? And do remember one point, that these releasing factors do not release the polypeptide. In translation, they do not release. The name is a misnomer. Which thing is provided for the release of polypeptide? For the release, it is peptidyl transferase ribozyme activity. I told you earlier also, right? Then in translation, in elongation, there is translocation. Translocation is movement of ribosome on mRNA. Be careful, it is not movement of mRNA, it is movement of ribosome on mRNA. One GTP is used in translocation, and this translocation requires factors, EFG factor, this EFG factor in prokaryotes, and elongation factor 2 in eukaryotes. Right? Now see this question. During translation, how many ATPs and GTPs are used to add one amino acid in the growing polypeptide chain? Here your answer is 2 ATP and 2 GTP. 2 ATP where? In the attachment of amino acid with the tRNA, right? That is charging of tRNA or activation of amino acid, right? Then 2 GTPs are used. 1 GTP in the elongation step for adding amino acid, and other GTP for translocation, the movement of ribosome. Okay, so total 4 high energy phosphate bonds are used to add one amino acid. So if a protein has 100 amino acids, then 100 into 4 is 400 high energy phosphates will be used to add all those amino acids in the protein. It is very high energy consuming pathway, right? Then our next topic is CRISPR. CRISPR-Cas9 system. What is the full form of CRISPR? CRISPR is clustered, regularly interspersed short palindromic repeats, right? So there's a palindromic repeat sequence present in the genome, right? And Cas9 is the enzyme. It is the enzyme. It is an endonuclease. It can cut CRISPR associated sequences. So this system is used to create double strand DNA breaks. This is a very, very new technology and advancements are going on in this. This is a very cheap and easy method to create breaks in the DNA. So CRISPR-Cas9 system is like a scissor with which we can cut the DNA, and that too with very cheap and easy methods. Before CRISPR, whatever methods were available were cumbersome also and costly also. But this system has now brought a revolution in molecular biology. So it is very, very important. Actually, this CRISPR system is an immune system, is naturally present immune system in bacterias against their viruses, against bacteriophages, viruses of bacteria, right? So using this system, bacteria can cut the viral genome and can protect itself from the virus. And this is stored as a memory in the bacteria, and this is transmitted to the next progeny. This memory is transmitted to the next progeny of the bacteria, so that many generations of bacteria are protected from that particular virus, right? So this CRISPR system is naturally present in bacteria, but it can be modified for use in molecular biology, for use in eukaryotes, right? For example, we recently used this system for COVID-19 testing, and the CRISPR COVID-19 test was given the name Feluda. Feluda full form is FN-Cas9, FN-Cas9 editor-linked uniform detection assay. Uniform detection assay, right? This system detects genomic sequence of SARS-CoV virus, right? It is more accurate and has quick turnaround time as compared to RT-PCR, which was used earlier, and it is a simple paper strip test. Simple paper strip test like a pregnancy test. So mass detection is possible through this because it has better ease of use as it is a simple test, so not a big lab is required. Okay, so this way CRISPR was used in COVID detection also. Also note that in CRISPR, for genome editing in CRISPR, which repair mechanism is used? Question was recently asked. It is non-homologous end joining repair is used. Non-homologous end joining repair. This repair method is usually used for double strand break repairs, right? The earlier repair methods which I told you, nucleotide excision repair, base excision repair, mismatch repair, they all were for single strand DNA break repair, right? Then our next topic is PCR. PCR is polymerase chain reaction. This is amplification of DNA. And the components required in PCR are, number one is double-stranded DNA, which is to be amplified, right? The double-stranded DNA, the source DNA, which is to be amplified. Number two, we require primers, 2 primers, one for each strand, right? Then we need the enzyme. The enzyme is Taq polymerase. Taq polymerase taken from bacteria Thermus aquaticus, because this bacteria can survive and withstand high temperatures. And in PCR, we use high temperature for the DNA strand separation. So we need such an enzyme which is stable in high heat. Okay. Then fourth is the substrate of this enzyme. The substrate is deoxyribonucleotides. Okay. We will never use dideoxyribonucleotides in PCR because if that is used, DNA synthesis will stop, right? Then fifth is we need magnesium ions because the enzyme polymerase require magnesium ions. So we take magnesium ions in a medium of buffer, right? These 5 components are required. Then there is one real-time PCR, real-time or quantitative PCR. See, the normal PCR is called end PCR. Normal PCR is called end PCR. Means when PCR will end, then we come to know that how much DNA is synthesized. Because the machine has high temperature and we cannot open the machine in the midway. Okay. So they developed this new one, real-time PCR or quantitative PCR. This can tell the quantity of DNA in real time during PCR, because in addition to the 5 components required for PCR, this PCR uses cyber green dye also. This cyber green dye gives fluorescence when it is bound to double-stranded DNA. So, as double-stranded DNA is getting amplified and getting increasing, the fluorescence is also getting increasing, which can be detected, and the quantity of DNA can be measured by the machine, and it displays on the machine that how much DNA is synthesized, right? So this was real-time PCR. Now we will do RT-PCR. RT-PCR stands for reverse transcriptase PCR, with the help of which we can amplify RNA also. Okay, so let's say we have some amount of RNA which we need to amplify. Okay, so first this RNA will be converted to DNA by reverse transcriptase enzyme. Okay, and this is called cDNA or complementary DNA. Now this cDNA will be amplified using normal PCR, that is polymerase enzyme. Okay, so we get lots of this cDNA, By transcription it can be converted to RNA, so we have lots of this RNA now. Okay, so in RT-PCR there is one enzyme, TTH polymerase. TITH polymerase or TTH polymerase. This polymerase has activity of reverse transcriptase enzyme also. It has activity of DNA polymerase also. So this is used in RT-PCR. Then our next topic is genomic imprinting. Genomic imprinting means inhibited. So the gene is inhibited. Transcription or translation will not occur, right? So how the gene is inhibited? Most common method of inhibition of gene in genomic imprinting is DNA methylation at CG site. Okay, this is frequently asked. So DNA methylation at CG site will inhibit the gene. What is CG site? These are two strands of DNA. C and G are together present on the same strand. Then this C is methylated. Okay, C of CG site is methylated. This will lead to gene inhibition, right? So this is the most common method. And other method is histone post-translational modifications. Histone post-translational modifications like histone methylation or deacetylation, right? This DNA methylation is detected by sodium bisulfite method, and post-translational modification of histone are detected by CHIP, C capital H small, I capital P capital. That is, CH stands for chromatin immunoprecipitation. Right? Then the diseases due to genomic imprinting. Diseases which can occur due to genomic imprinting. There are only two diseases. One is Prader-Willi syndrome, in which paternal allele of the gene, paternal allele is deleted and maternal allele is imprinted. Okay, so the gene will be affected because the mother's copy is imprinted, that is inhibited, and the paternal copy is deleted. So that full gene is not working, right? Now, in Angelman syndrome, maternal allele is deleted, and paternal copy of the allele is imprinted. So basically both the alleles are not available, so gene's function will be affected, right? Then next technique is microarray or chip. Now be careful, I have written chip in small letters, CHIP. Okay, this means a small chip is there in this technique where lots of DNA sequences can be placed and multiple mutations can be detected. Multiple mutations can be detected by this technique, microarray. Multiple mutations multiple gene expression analysis can be done. Comparative genomic hybridization can be done. So basically anything multiple, anything comparative is, if asked, go for microarray. And microarray can also detect SNPs. What is SNP? Single nucleotide is variable in different population. That is single nucleotide polymorphism. Okay, but microarray cannot detect aneuploidy, that is monosomy and trisomy. So our next technique, that is karyotyping. Karyotyping is best for detection of monosomy and trisomy. Okay, but karyotyping can be done only in metaphase arrest, right? So next technique, FISH, it can detect aneuploidy. It is even better than karyotyping. It can detect aneuploidy and it can be done in any phase of cell cycle, right? But if they ask you best technique for aneuploidy, that is karyotyping. And FISH is fluorescent in situ hybridization, which can detect aneuploidy and it can be done in any phase of cell cycle, and it is a very fast and rapid technique. Results can be obtained within 24 hours. And FISH also tells the location of gene on chromosome. Location of gene on chromosome. This is frequently asked. And FISH can also detect microdeletion, small changes like microdeletion, amplification, complex translocation. Okay. On the other hand, karyotyping is time consuming also, and it cannot be done in any phase of cell cycle. And in karyotyping, the whole chromosomes are seen in line at the equator. So these small changes like microdeletion, amplifications and complex translocations cannot be detected, right? So next topic in molecular we are going to do is Mendelian inheritance disorders, right? So Mendelian inheritance disorders, these are single gene disorders. There is defect in single gene. Okay, so here we all know we have autosomal recessive, autosomal dominant, X-linked recessive, X-linked dominant and Y-linked disorders. Okay, so the important ones which you need to learn, only that I am going to take in this rapid revision. Only one example of autosomal dominant you have to do, that is familial hypercholesterolemia. Familial hypercholesterolemia or type 2A cholesterolemia, right? Hyperlipoproteinemia, right? Then most of your biochemical disorders are autosomal recessive. So we can note down all of these. So here we have all mucopolysaccharidosis like Hurler disease. So all mucosaccharidosis except Hunter disease, because that is X-linked recessive, right? Then all urea cycle disorders except OTC, that is ornithine transcarbamylase deficiency. So this is also X-linked recessive. Then we have all sphingolipidosis like Niemann-Pick disease, Gaucher disease, right? Except one, that is Fabry's disease. Okay, Fabry's disease, that is also X-linked recessive. So these three are X-linked recessive, right? Then other autosomal recessive disorders are all the amino acid disorders like alkaptonuria, albinism, phenylketonuria, right? Then we have all glycogen storage disorders, right? Then orotic acid urea, Wilson's disease, ADA deficiency, ADA deficiency and hemochromatosis. Hemochromatosis, right? So now we can write other X-linked recessive disorders. 3 I have already told you on the previous slide, right? So here we have Menkes disease of copper deficiency. Then we have Lish-Nyhan syndrome with self-mutilation, right? And we have G6PD deficiency with hemolysis and Heinz bodies. Okay, now you can do some important pointers to determine inheritance patterns. So if a question is given to you and the family history is given and they say that both male and female affected with equal frequency, both male and female affected with equal frequency, then it is autosomal. It is autosomal, right? Then if at least one parent is affected with the disease, if at least one parent has the disease, then it is dominant pattern of inheritance. Then if they say that if neither of the parent has disease, neither of the parent has the disease, then that means it is recessive inheritance pattern, right? If they say that if all sons are affected, father is affected and all sons are affected, then it is Y-linked inheritance. Then if they say mother affected and all offsprings affected, mother affected, all offspring affected, that means it is mitochondrial inheritance, right? Then if they say there is no male to male transmission, no male to male transmission, then it is X-linked because this disease is transmitted from mother, right? Then if they say in some disease more males are affected, and these affected sons are born to unaffected mother. Unaffected mother means mother is the carrier here. So most probably it is X-linked recessive inheritance, right? So with these pointers you can solve the questions. Then we have to understand one thing, that is clinofragment. 2-3 times question is asked, right? So we know that DNA polymerase 1 enzyme is involved in DNA replication and it has three activities. One is it has primer removal activity, that is 5 to 3 exonuclease activity. Then the synthesis activity, that is the polymerase activity, the normal one, that is 5 to 3 polymerase. Then we have proofreading activity also in this enzyme, which is 3 to 5 exonuclease activity, right? So here this is represented from N-terminal to C-terminal. The sequence of these segments are represented on this diagram in the DNA polymerase, right? So we have 5 to 3 exonuclease activity on N-terminal side and other 2 activities on the carboxy-terminal side, right? Now with bacteria subtilis, Bacillus subtilis, there is a toxin subtilicin which can cut here. So we get a smaller fragment on the left side and the larger fragment on the right side which we get is known as clinofragment. So you can say that clinofragment is formed from DNA polymerase by removal of 5 to 3 exonuclease activity, right? Now check this thing, that severity of damage in the mutation is frequently asked. So we have many point mutations. So the severity of damages, that frame shift mutation is most severe because here the whole frame of codons is shifted and many amino acids down the line can be changed. So this is most dangerous. In frame shift mutation, the nitrogenous bases are added or deleted which are not in multiple of 3. That's why codon is shifted, right? Whole frame is shifted. Then is nonsense mutation, which is less dangerous than frame shift. In nonsense mutation, some amino acid is replaced by stop codon and there is early termination of the protein. So a shorter protein will be synthesized, right? Then comes missense mutation, in which one amino acid is changed with another amino acid, right? So this is less dangerous than the nonsense mutation, right? So this is it with molecular biology. Now we will be starting miscellaneous. So students, let's do this last topic in rapid revision, that is miscellaneous. Okay. So small topics I will be taking under this, like vitamins and plasma proteins, etc. So let's start with RQ. Okay. RQ is respiratory quotient. Respiratory quotient. Many questions are asked on this. Okay. So what is RQ? RQ is amount of carbon dioxide produced, divided by amount of oxygen used during metabolism. Okay. Now for carbohydrates, oxygen used and carbon dioxide produced is same, that is 6. That's why RQ for carbohydrates is 1. Okay. Then RQ for proteins is 0.8. Look at P of protein. This P can be converted to number 8. Okay. So this way you can learn. Then fats RQ is 0.7. F of fat can be converted to number 7 like this. Okay. F of fat number 7. Okay. Then RQ for mixed diet. For mixed diet is 0.85. RQ for alcohol is 0.66. And if they ask you that carbohydrate rich diet is taken or excess carbohydrate diet is taken, then what is RQ? Then you will say RQ is more than 1. More than 1, usually 1.2. Okay. And they ask you that what happens to RQ in diabetes. In diabetes, what happens to RQ? See, in a normal person is considered in fed state. So the main fuel for body is carbohydrates, right? And in diabetic person, it is considered that the body is in fasting state because cells are not getting glucose due to low or deficient insulin, right? Then the state is considered fasting state, okay, diabetic state. So here in fasting state, fats are the main fuel, right? Now RQ, what happens to RQ in diabetes? So the main fuel is shifted from carbohydrates to fats. So RQ decreases because RQ of fat is less than carbohydrates, right? So RQ decreases in diabetes. And on giving insulin, what will happen? Glucose will again enter the cells and the body will be again in fed kind of state. So RQ will increase on giving insulin in a diabetic patient, right? You can also note that RQ decreases in diabetes and alkalosis. Alkalosis also RQ decreases. Then note down RQ increase in which condition? RQ increase in acidosis. In acidosis, during fever and during exercise, RQ increases, right? Then next we will do collagen. Collagen is a glycoprotein. It is most abundant of all human proteins. The primary structure of collagen is glycine XY repeated again and again, repeated n number of times, right? So we can say that every third amino acid is glycine, right? Every third amino acid is glycine. And X and Y, they can be proline, lysine, hydroxyproline or hydroxylysine, right? Then what are the types of collagen? There are around 28 types of collagen present in the body and only few which are important that we will be doing. So type 1, 2, 3 and 4. Okay. Within mnemonic, SCAB, SCAB. Okay. S for skin, Type 1 is present in skin and it is most abundant type because skin is the largest organ, right? Then C for connective tissues. Type 2 present in connective tissues. Type 3, A for arteries and cardiovascular system, right? And this type 3 is also the main form which is present in healing and granulation tissue. Present in healing and granulation tissue, right? This question was asked. Then type 4 is present in basement membrane of glomerulus in the kidneys, right? And this type 4, if defective, this will lead to Alport syndrome, in which there is hematuria, blood coming in the urine, and it can if not treated, then it can lead to end-stage renal disease, right? So these were 4 types of collagen. Then one more type which is important is type 7. This is present at the junction of dermis and epidermis, right? And there is defect in type 7. This leads to epidermolysis bullosa, in which there are skin blisters. There are skin blisters like this because the junction of dermis and epidermis is now loosened due to absence of this collagen. Then let's do PTMs of collagen. PTM of collagen, that is post-translational modification. Okay. First is hydroxylation. With hydroxylation, there are formation of numerous hydrogen bonds in collagen which make this protein strong, right? Hydroxylation occurs for proline and lysine, and the enzymes are prolyl hydroxylase and lysine hydroxylase or lysyl hydroxylase. Then for lysine it is lysyl hydroxylase, right? And both these enzymes require iron and vitamin C. So remember, vitamin C has a role in collagen formation. That's why deficiency of vitamin C leads to scurvy, in which there is loosening of all the strength of collagen, right? Therefore, this leads to bleeding gums. Now, second PTM of collagen is glycosylation, that is adding carbohydrate. When this carbohydrate is added, then the bond which is formed is aldol condensation, which is intramolecular crosslinks in collagen. Again, they give strength to the molecule. And the enzyme responsible here is lysyl oxidase, which requires copper, right? So in collagen, iron and copper, these 2 minerals are required, and 1 vitamin, that is vitamin C, is required. Okay. Then let's do plasma protein, that is transthyretin. It was earlier named as prealbumin because it was moving ahead of albumin in electrophoresis. That's why that name was given depending upon its electrophoretic movement. But now the new name is more specific to its function, that what this protein is doing. So it is doing transport of written in the name because it is helping in the transport of thigh and retin. Transport of thyroxine hormone and retinol binding protein, right? That's why the name, that is transthyretin. Okay. And this protein is present in blood as well as CSF. Okay. Now another important protein, plasma protein, is ceruloplasmin. Ceruloplasmin helps in the transport of copper, right? As a precursor, apoceruloplasmin is formed in liver. Apoceruloplasmin is formed. Then it joins with approximately 6 copper atoms, and then it is ceruloplasmin, short form CP. Okay, ceruloplasmin is formed, and this goes in blood and takes copper from liver to the peripheral tissues, right? And this enzyme also has ferrooxidase activity. It converts Fe2 to Fe plus 2 to Fe plus 3, right? And this is required for iron transport in the body. Okay, so this ceruloplasmin is also very important in iron metabolism. Menkes and Wilson's disease. It is very, very important. So let's get started. Menkes and Wilson's. I will help you learn few points here on this slide. So see here, Menkes, M alphabet comes before, and then W for Wilson's, W alphabet comes afterwards. So very easy to learn that M for Menkes is copper deficiency, less copper in the body, and W for Wilson is more copper excess in body. And very easy to do next point also alphabetically. Here defective protein is 7A protein. So A comes before, then B comes afterwards. In Wilson's, the defective protein is ATP7B protein, right? Very easy to learn. Okay, let's do the other differences now. Menkes disease is X-linked recessive. Wilson's disease is autosomal recessive. Then the protein which is deficient in Menkes disease is present in intestine, and in intestine its work is absorption of copper, right? So absorption of copper will not be done here, and there will be copper deficiency in body, decreased copper in blood and urine, and ceruloplasmin. Ceruloplasmin is the protein which transports copper, and it is formed in liver. This ceruloplasmin will also be decreased because there is not enough copper to be incorporated in ceruloplasmin, right? Then Wilson's disease. The protein defective is ATP7B protein. It is present in liver. It has two functions. One is to throw copper out of body via bile. So this won't be done now. Copper is not thrown out of body via bile. So there will be copper increased in liver and copper excess in body, right? Then the other function of this protein is to incorporate copper in ceruloplasmin. CP stands for ceruloplasmin. Okay. So now copper is not getting incorporated into ceruloplasmin. Although there is lot of copper in the body, but it is not getting incorporated into the ceruloplasmin. So ceruloplasmin active protein or holoprotein is not getting formed. So ceruloplasmin is decreased, right? So there are many differences between Menkes and Wilson's, but there is one thing common in both of them, that ceruloplasmin CP protein is decreased in both, right? Then let's do these two diseases in detail one by one, and here in this video I will be showing you many images which will help you in solving image-based questions. So first let's take detail of Menkes disease. In this, the signs and symptoms are premature birth, hypotonia, growth retardation, and mental retardation also occurs in these patients. And these patients can be easily recognized by grey depigmented hair. You can see in all these pictures depigmented hair, right? And why these depigmented hair? Because enzyme tyrosinase is affected. Enzyme tyrosinase is an oxidase, and any oxidase requires copper. Okay, so because copper deficiency is there in Menkes disease, so this enzyme won't be working properly, and this enzyme is responsible for making pigment melanin, right? So therefore it is affected. Then brittle kinky hair. They are very brittle, weak hairs. You can see in all the pictures, very brittle hair, right? Why this? Because collagen is affected. One of the enzymes required in collagen synthesis is lysyl oxidase. Again oxidase, so it requires copper. In copper deficiency, this enzyme is not working properly. So there is not enough strength in this protein collagen, right? That's why brittle, kinky hair. Because of this thing, Menkes disease is also known as Menkes kinky hair syndrome. Kinky hair syndrome, right? Then obviously there will be decreased copper in blood and urine in these patients, right? So this was about Menkes disease, right? Then Wilson's disease, there is increased copper in blood and urine. Then copper is increased. First, copper is increased in liver, and due to copper's toxicity, liver damage will occur. Once copper is increased in liver, after that it will move from liver to extrahepatic tissues, and wherever it will go, it will get deposited and it will cause damage and various signs and symptoms. So brain will be affected and it will lead to neurological degeneration. Then copper deposition in kidneys will lead to renal damage and urolithiasis. Then copper damage in bone marrow and RBCs. This will lead to hemolytic anemia. Then copper deposition in eyes. It will lead to sunflower shaped cataract and KF rings. So let's see these two pictures on next slides. First see sunflower cataract. Sunflower cataract, this kind of picture if given, so you can recognize. So this occurs in Wilson's disease. Then Kayser-Fleischer rings. Kayser-Fleischer rings. What are these? These are green or golden brown. So you can see green color here, golden brown color here. This is due to deposition of copper. It is seen in the descemet membrane of cornea, right? So this is Kayser-Fleischer ring in Wilson's disease. Then write down what is the treatment of Wilson's disease. Treatment of Wilson disease is giving penicillamine, because penicillamine chelates copper, right? Then there was a recently asked question. You just read the question. In which of the following disorder ATP7A protein is deficient? So you know it is Menkes kinky hair syndrome, right? And in Wilson's hepatolenticular degeneration, see, Wilson is also known as hepatolenticular degeneration. In this, ATP7B protein is deficient, right? Now next let's do vitamins. Vitamins, quick one-liners we will do in vitamins. First, let's do the basic that there are water-soluble vitamins, B complex and C. Water-soluble vitamins act as coenzymes in the body, right? And water-soluble vitamins are not stored in the body, usually not stored in the body, but one exception, that vitamin B12 gets stored in the body in liver, right? Now, on the other hand, we have fat-soluble vitamins, that is vitamin A, D, E and K. Usually, they are stored in the body, so they can have toxic effects also if they are taken in excess, because they are stored in the body. And they don't act as coenzymes usually, but one exception, that vitamin K is that fat soluble vitamin which act as coenzyme in the body, right? And there is one more exception, that vitamin K is the fat soluble vitamin which has a water soluble form. It has a water soluble form, that is menadione. Menadione, also called vitamin K3. This is not natural form of vitamin K, it is synthetic form of vitamin K used therapeutically, right? Then note down vitamins which are synthesized by bacterial flora in the intestine. These are vitamin B2, B5, B7 and vitamin K. Okay. Then we have atypical vitamins. Atypical vitamins, those vitamins which can be synthesized by human enzymes. They are vitamin D and vitamin B3 niacin. Okay. Then there are two vitamins which are not present in vegetarian diet. Two vitamins not in vegetarian diet. They are vitamin D and vitamin B12. So in strict vegetarians, vitamin D and vitamin B12 deficiency can occur, right? And in strict vegetarians, B12 deficiency will be more than vitamin D deficiency, because vitamin D can be obtained from sunlight also, right? Then note down vitamin deficiencies which are having neurological signs and symptoms. Having neurological signs and symptoms. These are vitamin B1 because it causes beriberi, vitamin B3 in which there is dementia, delirium, etc. Then there is vitamin B6 where neurological symptoms are found. Then vitamin B12 and vitamin E. Both vitamin B12 and vitamin E, they have peripheral neuropathy. But how to distinguish? B12 deficiency will be having megaloblastic anemia also, but in vitamin E deficiency, there will be hemolysis, right? Then note down site of absorption of vitamin B9. Site of absorption of vitamin B9 is duodenum and jejunum. Then site of absorption of B12 is ileum. Okay, and this absorption of B12 also requires intrinsic factor IF. Deficiency of intrinsic factor leads to pernicious anemia, right? Then let's do vitamin deficiency anemias. Vitamin deficiency anemias, they can be of two types. One is microcytic hypochromic anemia, which can occur due to vitamin C deficiency or vitamin B6 deficiency, right? Because vitamin C is required for absorption of iron, that's why heme synthesis will be affected, and vitamin B6 is a coenzyme for the rate limiting step of heme synthesis, that is ALA synthase. Okay. Now the other anemia which can occur due to vitamin deficiency is megaloblastic anemia. This can occur due to vitamin B9 or B12 deficiency. Okay, and there are no neurological symptoms in B9 deficiency, but there is peripheral neuropathy which is progressive, that is found in B12 deficiency. Then a question was asked, a clinical question was there, that a person consumes raw eggs, and one day he fell down, so he had hypoglycemia. What is the reason? Look, raw eggs, they have a protein named avidin. This tightly bounds with vitamin B7 or biotin, and it will not allow the absorption of biotin. So there will be biotin or B7 deficiency in this person. B7 is important coenzyme for carboxylase enzymes, right? And pyruvate carboxylase is the first step in gluconeogenesis, right? So this is the reason that raw egg consuming person will be having hypoglycemia, right? Then something about vitamin A, the active form is retinol. So it is absorbed from intestine in the form of retinol. Then it reaches liver, it is stored in liver in the form of retinol esters, and approximately 1 year supply of vitamin A is usually stored in liver. In which cells? In ITO cells, also called perisinusoidal cells, right? And in liver, this retinol ester is joined to retinol binding protein, RBP, retinol binding protein. This retinol binding protein is synthesized in liver, right? Then when from liver this vitamin A has to go in circulation, then a ternary complex is formed. Ternary means 3. So three things are joined here: retinol ester plus retinol binding protein plus transthyretin. There is a complex of these three things. Why this complex is made? So that vitamin A is not lost in urine. If we only have retinol ester and retinol binding protein, then that molecule is having low molecular weight. It can be filtered out through kidneys in urine and will be lost from the body. So transthyretin helps in maintaining it in the body. Then what are the signs and symptoms in vitamin A deficiency? Vitamin A deficiency is the most common cause of preventable blindness. Right? So eyes are affected mainly because vitamin A is involved in Wald's visual cycle. Right? Now, first there is loss of sensitivity to green light. Loss of sensitivity to green light. Patient has night blindness. Night blindness. Bitot spots. Bitot spots. All these images which are clinical images required for your exam, they are covered in snapshot video. So do watch that video also, right? So there are bitot spots, keratomalacia, then follicular hyperkeratosis, then corneal ulceration. Vitamin A deficiency patients have frequent UTI and respiratory tract infections. Then other is xerophthalmia and immunosuppression. That's why these frequent infections are occurring, right? So this was deficiency of vitamin A. Now excess vitamin A will lead to vitamin A toxicity, right? And this leads to pseudotumor cerebri, a condition which resembles brain tumor. There is increased intracranial tension and headache in this patient, right? Then vitamin D. Synthesis of vitamin D is very important as it occurs in body. So the organs which are involved are, first, with the help of UV rays, the synthesis of vitamin D starts in the skin. Then in liver, one reaction occurs with the help of enzyme 25-hydroxylase, right? And then in kidneys, the rate limiting step occurs with enzyme 1-alpha-hydroxylase. Okay? And the synthesis of vitamin D starts from cholesterol. Okay? And to be more precise, it is 7-dehydrocholesterol, right? And the active moiety of vitamin D is calci-triol. Tri means 3, ol means OH. It has 3 OH groups, and these 3 OH are at position number 1, position number 3 and position number 25 in cholesterol, right? Then vitamin D deficiency. Vitamin D deficiency will lead to rickets in children, where there is rachitic rosary, that is beaded appearance in the ribs in the chest. Then hot cross bun appearance of head due to non-closure of fontanelle. Then they have knock knees, bow legs, right? So this is rickets. Then in adults, there is osteomalacia in adults, and these adults have soft bones leading to frequent fractures, and they usually have waddling gait, right? So this was deficiency of vitamin D. Now excess of vitamin D will lead to hypervitaminosis D, in which patient will be having hypercalcemia. Excess of calcium will be there. Hypercalcemia, hypercalciuria, due to which there will be renal calcium stones, and deposition of calcium in soft tissues also. like blood vessels. There will be calcium deposition in blood vessels, and blood vessels will be rigid, leading to hypertension. So there will be hypertension, increased blood pressure in hypervitaminosis D. Then vitamin E. Vitamin E is most potent lipid phase antioxidant, right? Other antioxidant vitamins are vitamin C, vitamin A, and vitamin D. So these four are antioxidant vitamins, okay? E, C, A, D, right? Now, vitamin E works synergistically with selenium, glutathione, and vitamin C in its antioxidant role, okay? I repeat, vitamin E works with selenium, mineral, glutathione, and vitamin C for its antioxidant role, right? And deficiency of vitamin E will lead to peripheral neuropathy but no megaloblastic anemia. Here patients have hemolysis and ophthalmoplegia. The question was asked that which vitamin deficiency leads to ophthalmoplegia, right? Then vitamin K. I told you vitamin K is the only fat soluble vitamin which has a coenzyme role. It has coenzyme role in the activation of clotting factors 2, 7, 9 and 10. These clotting factors, they have glutamate in them. Glutamate has carboxy group, right? Now vitamin K will do more carboxylation of these glutamate residues in the clotting proteins, making gamma carboxy glutamate. And here vitamin K is working. So hydroquinone is the active form of vitamin K which is involved here. So this gets converted to epoxide form of vitamin K. Here oxygen and carbon dioxide are added. The glutamate is carboxylated and vitamin K is oxidized. Okay. So here enzyme is vitamin K dependent carboxylase. Right? Now epoxide form is converted to quinone form by adding one hydrogen, and the second hydrogen added and hydroquinone form is synthesized. So here enzyme is epoxide reductase, which is inhibited by drug warfarin, which act as anticoagulant. Okay, and here the enzyme is quinone reductase, right? So now we will do vitamin K deficiency. In vitamin K deficiency, there will be easy bruising, bleeding tendencies, increased prothrombin type, and hemorrhagic disease of newborn can occur, especially in preterm infants. There are many reasons of hemorrhagic disease of newborn. One, that the intestine of newborn is sterile. Bacteria are not there to synthesize. Number two, there is poor placental transfer of vitamin K. Also, breast milk is poor in vitamin K. And hepatic immaturity. The liver of newborn is also not having good stores of vitamin K because it is immature, right? Then in vitamin C deficiency also bleeding occurs, and there is increased bleeding time in vitamin C deficiency, right? Vitamin K deficiency patients are usually patients which are taking long-term antibiotics or fat malabsorption patients, right? Vitamin C deficiency patients are usually those patients in which diet is poor in citrus fruits or fresh fruits and vegetables, right? This way you can differentiate. Then some more one-liners we can do. Cyclic GMP is also a second messenger involved in few things in the body. One, it is involved in vitamin A visual cycle. Then it is involved in the nitric oxide vasodilatory effect, and it is also involved in the activation of ANP and BNP natriuretic peptides, right? Then frequently asked question is transparency corneal transparency is due to which GAG, which glycosaminoglycan? It is keratin sulfate, right? Then which lipid deficiency causes retinitis pigmentosa? Retinitis pigmentosa. The answer here is DHA, docosahexaenoic acid, right? Then ApoB48 protein is synthesized from ApoB100 gene in intestine. So this is formed due to a process known as RNA editing, very important, which is a post-transcriptional modification. It is a post-transcriptional modification, right? Then HbA1c. Recently 2-3 times it is asked that what is the best method of detecting HbA1c? Answer is ion exchange chromatography, right? Then best investigation for any inborn error of metabolism. IEM stands for inborn error of metabolism. Best investigation is mass spectrometry. Okay, you can learn these one-liners so that recently asked questions are also covered here. Then buffer. A buffer is most effective when pH is equal to pKa, right? And buffering range, maximum buffering range if asked, then pH is equal to pKa plus minus 1, right? And minimum buffering is at which point? When pH is equal to isoelectric pH, right? Then major extracellular buffer is bicarbonate buffer. This is also considered an ideal buffer. It is considered an ideal buffer because both components of bicarbonate buffer, bicarbonate and carbon dioxide, they both can be altered for maintaining the blood pH. That's why it is considered ideal buffer, right? Then major intracellular buffer or urinary buffer is phosphate buffer. It is phosphate buffer, right? So this is it with your rapid revision. Many best wishes to you, and I also suggest that do watch snapshot video also for the images, right?